metabolites, lactose, lactate, valine, and proline. And in each square, what this is indicating in the upper right triangle, this is the expected abundance of that metabolite, and this is the observed abundance in that metabolite. And so for lactate, for example, there are several different pairs of ASF species, so of that Shadler flora microbe, where the amount of lactose is less than expected. So it's consuming that metabolite, lactose, more than you would anticipate from the data in the monoculture. For some metabolites like valine, you see transitions between sometimes the metabolite is produced more than expected, sometimes it's consumed more than is expected. Now this, let me just summarize the data, kind of this overview, and then go to a specific example of what we're talking about here. Of 1,290 metabolite co-culture groups for which we could predict some level of the metabolite abundance, the null hypothesis was rejected in 500 of those. So about 40% of the metabolite co-culture data is different than you would expect, suggesting that co-culture truly alters metabolism of a significant fraction of these metabolites. So let me dive in a little bit more specifically here. We have here, of the metabolites, the green dots represent different metabolites. We have across all the ASF pairs, these are metabolites where they're more abundant than expected. And on this axis is the statistical significance, so a decreasing p-value. So we have some metabolites that are more abundant than expected, others that are less abundant than expected. And this is of the subset of metabolites for which in the monocultures, they were produced by one species and consumed by another. So what's the interpretation of that? Well, for metabolites that fall into this category, there's a couple different hypotheses here. Perhaps the one species is consuming it more, or perhaps the other species is producing it less. For metabolites in this category, perhaps the one is consuming less and the other is producing more. You can similarly generate these different kinds of mechanistic hypotheses for the collection of data that you have about how the abundance of the metabolites are different in the co-culture than you would expect as a sum of the monocultures. So I'm gonna give you a very specific example. Two different microbes here, and that Shadler flora have a numbering system, which you can look up if you like, but there was a particular pair for which there was evidence that alanine and proline were being produced by one species and consumed by another. Now, and that the consumer of those metabolites had improved growth than would be expected, okay? Now, what's interesting about this is a classic pathway associated with stickland fermentation in clostridial species. So the survey of, again, expected versus observed metabolite levels suggested that 519 was a producer of metabolites that are frequent substrates for stickland fermentation, for which this other microbe could consume for a growth benefit, okay? Now, that's what that survey of the different metabolite levels, running it through that ConYE test allowed for. So we wanted to test that. And the experiment we did, we have here, this is the relative density relative to supplement, so how much growth this particular microbe is exhibiting under a few different media conditions. So this is just in the baseline media. And this is when we provide proline, alanine, isoleucine, and valine alone. And there's no statistically significant difference in the growth benefit. However, when we provide the proline and alanine together, there's a significant growth benefit for, again, 356, when those two metabolites are provided together, where there is no difference when other amino acids are provided in other pairs. So this suggested to us that this stickland fermentation was indeed facilitating growth of this ASF species 356. Okay, it wasn't just the availability of the individual metabolites, but providing those metabolites as a pair. Okay, that's one story. This was published. I'll point you to that paper. What I want to do with the remaining time that I have is talk a little bit about this next challenge. So to do this, let me first just give a little background on some metabolic network modeling that my lab and others here attending the symposium are very engaged in. And just to give a brief introduction to some of the fundamental basics for metabolic network modeling at a genome scale. First step often involves mapping genes to associated proteins and reactions that those proteins or enzymes will catalyze. This allows you to delineate what are the functions that the genome captures from a metabolic functionality perspective. Second step involves constructing what's called a stoichiometric matrix. This is mapping the stoichiometric coefficients for the substrates and components of a network as rows, and then in columns, the different transformations that convert substrates to products. Once we have that matrix formalism in place, there are a variety of different matrix linear algebra tools, linear programming, other optimization strategies that allow you to generate a variety of different kinds of predictions. Now, for this kind of framework, we often assume that there's equal access to all the functionality in this network, okay? But we know that's rarely, if ever, the case in biology. So methods have emerged that allow us to integrate expression data to delineate the functions that are active in one state or one cell type versus another. Some of these methods have been developed by people here at this symposium. Now, these approaches are readily extended with high quality metatranscriptomic data, so transcriptomic profiling data in microbiomes, where we can, when that transcriptomic data is of sufficiently high quality, where we can assign from a large pool of transcripts which transcript level is associated with each species in a complex system, in a complex community, okay? But many of those methods involve delineating what are the exchanges or what's allowed to come in and out of that particular species or of that particular cell. But in microbial communities, we have a pool of shared metabolites. And so it can become difficult to constrain which are the substrates that one species is consuming and another's producing, given this pool of shared metabolites. So a lot of expression data integration algorithms require that you define those kinds of constraints, but this can be difficult to do. So in part, to help address this challenge, Matthew Jenner, you see his picture in the bottom right there, he's a postdoc in my lab. He led the development of this approach we call Riptide. Now, I'm gonna point you to the paper. This just came out recently for the details, but let me just give you kind of a sense for the method. So this method does not require that you constrain those exchange fluxes. Instead, what it does is two key components. So one, this method takes transcript data, and where this blue line represents the abundance of transcripts, where the red line represents a weight that you're going to put on the corresponding reaction. So simple idea, if the expression level is particularly high, then we're going to allow for flux through that particular reaction. If the transcript levels are really low, then we have a strong preference to not push flux through that reaction, okay? Now, other methods have done this. Important development with Riptide is that we also pair it with an assumption of parsimony of the network enzyme usage. And the evolutionary argument here is that the cell isn't going to be using energy to express things that it doesn't need to use, okay? So this integration of mapping transcript abundance with assumption of parsimony allows you to reduce some of the assumptions that typically go in these gene expression integration algorithms. Now, just to give you kind of a graphical illustration of this method, let's say, for example, we have a cell and there's two different pathways here, two different pathways of metabolism. Glycolysis is just a couple of steps, and that's one option where with the consumption of glucose, we can produce ATP. But an alternative pathway might be something like stickling fermentation, where I'm consuming two different substrates to generate or satisfy my ATP demand. Now, this pathway is less parsimonious. Okay, there's more steps involved, more enzymes that would need to be synthesized. But if my transcript data indicates that these are highly expressed in comparison to the others, I can break that parsimony so that this becomes my prediction of a metabolic state. Okay. Now, I'll just point you again to that paper from that on which Matt is a first author. There's several different analyses that were done to illustrate, offer some validation and confirmation of the validity of the approach. This is some example data where he integrated with riptides in an E. coli model, some gene expression data associated with LB aerobic, some defined media, both aerobic and anaerobic, defined minimal media, and where we predict genes that are essential, non-essential, et cetera. And the categories of genes that show up as essential and non-essential are as you would anticipate, given the different media conditions that they have. And there's additional validating information that I'll point you to the paper to help understand. Now, most recently, for some unpublished work, Matt has been extending this approach, again, back to a microbial community that has some clinical relevance. And his focus has been on understanding Clostridioides difficile. Now, this is a bug that is just a tremendous healthcare burden in the United States, 500,000 cases a year, approximately $5 billion healthcare costs associated with this. And infections arise when there's a disruption of a healthy gut flora. People are taking antibiotics, wipe out a lot of the healthy bugs that are in the gut. C. difficile will thrive in that environment. Now, there's a lot of interesting recent information that's coming out about the role that metabolism plays in the process of infection with C. difficile. And I'll just point you to this recent preview that Matt wrote up that was posted in Cell Host Microbe just a couple weeks ago, where he summarized a couple of recent papers, which was the actual interesting discovery and work, demonstrating how C. difficile capitalizes on the host environment metabolically in order to facilitate the process of infection. So that preview, I think Matt did a good job summarizing some of that recent work. So Matt constructed a recent genome-scale model of C. difficile. There have been a handful of others, but just to summarize, he was able to curate around, and I just included a list of a variety of different metabolic pathways that he curated in his recent reconstruction. He also ran through a variety of different validating data on gene essentiality, as well as some growth data that Matt has been able to compare model predictions with, just again to demonstrate the quality of the C. difficile model that he had built. Now, let me integrate this brief introduction, this new C. difficile model with the riptide approach for expression data integration in a clinically relevant setting. So in collaboration with a colleague, Rita Tamayo, who's at University of North Carolina in Chapel Hill, she recently discovered this interesting phenotype with C. difficile, where under certain growth conditions, and she did this, there's this beautiful paper that she published in PLOS Biology last year, where she demonstrated C. difficile undergoes this phenotypic switch. And there's these variants in a culture between what's called smooth and rough, as you see here, just because of the morphology. And she did a lot of work to demonstrate how certain genes were important in that those phenotypic variants, as well as some of the implications in the pathogenesis of C. difficile because of this phenotypic switch. But one question that she had in our conversations with her was, what's the role of metabolism play in this setting? And so Matt took his new C. difficile model and integrated some gene expression data that Rita had of the smooth and rough variants. And again, as you can imagine, expression data from some of these colonies or in these complex communities can be a lot easier to generate than metabolomics data specific to each variant. So real quick, and this is my last data slide here, Matt integrated with Riptide the expression data associated with these two variants. Okay. First of all, the predictions of the respective growth rates were comparable to what's observed experimentally, and that there's no difference in the growth, which is as observed experimentally. Matt then took those models to predict genes that were essential in one variant versus another. And there's an interesting observation he made is the role that glucose as one substrate and proline as another substrate had. Genes associated with glucose were essential in smooth variant, and genes associated with proline and Stickland fermentation related pathways were essential in the other. So similarly, Matt had predictions with his Riptide models for uptake rates of these two different substrates that match. Now, this is a picture that Matt just generated just a few days ago, just to emphasize that this prediction of how critical glucose is as a part of these two different phenotypes. In the absence of glucose, smooth colonies take a more rough shape. You see the more finger-like extensions on the colonies. Matt's going to be repeating this data. Again, this is just brand new in the last few days. But again, to emphasize that this important variant that has important pathogenicity implications of smooth versus rough variants of the C. difficile cultures, that the model helped us point to essential metabolic processes that were critical as a part of this phenotypic switch. So in summary, talked about this ASF community as a model microbiome, some of this work to characterize metabolites that are shared, this new method for gene expression data integration, and how some of these approaches are allowing us to really drive discovery activities and understanding important pathogens associated with microbial communities. I'll just give a little plug here of some training training opportunities at University of Virginia, which is a wonderful place. Grad student applications are coming due in the near future. We have great training in systems biology at University of Virginia, which has a medical center and engineering school and close walking distance between each other. So let me acknowledge those involved. Greg Medlock, again, is a PhD student that drove that one. Project's now faculty in pediatrics at UVA. Matt is a postdoc in the lab who drove a lot of the other work that I talked about. The funding that supported the work I mostly focused on here was from the NIH. And then you see some links to our Twitter account and everything else. So Priyanka, Ines, I took like 1 or 2 minutes longer than I was supposed to. I'll stop talking now.
Thank you so much, Jason. This was really enlightening talk. And your group is doing amazing work in the field of microbiome also. This is amazing. And we have a lot of questions from the participants, but we'll take them at the end of the session. I would now like to introduce Ines, and if you would like to share your screen. So for the second talk in this session, we have Professor Ines Thiele. She is the principal investigator of the Molecular Systems Physiology Group at the National University of Ireland, Galway. Her research aims to improve understanding of how diet influences human health. Ines has been pioneering models and methods allowing large-scale computational modeling of the human gut microbiome and its metabolic effect on human metabolism. And Christian showed the Agora models yesterday during the course. Those models were developed in Ines's group. So her group has been doing phenomenal work in this field. Just to give a brief background of Ines's academic profile, she earned her PhD in bioinformatics from the University of California, San Diego in 2009. From 2009 until 2013, she was an assistant professor at the University of Iceland. Later, she joined as an associate professor at the University of Luxembourg. And since Feb 2019, Ines is a professor for systems biomedicine at the National University of Ireland, Galway. She is the recipient of several awards like the Attract Fellowship, Embo Young Investigator Award, and she has also got the prestigious ERC Starting Grant. She's an author of more than 90 international papers, so she has done amazing work, and reviewers for multiple journals and funding agency. She's particularly interested in applying her computational modeling approach for better understanding of inherited and neurodegenerative diseases. Without further delay, Ines, I would like to welcome you to speak on in the symposium.
Thank you very much, Priyanka, for the extensive introduction. So that means I don't have to introduce the purpose of the lab anymore that detailed. Jason, good to hear you. And it's all right for the 5 minutes or 2 minutes, whatever it was. It's a great pleasure to talk to all of you and to introduce our work. Particularly the most recent work we've been doing in the laboratory at the National University of Ireland, Galway. And as Priyanka said, the focus of the lab is really to understand how diet influences human health. And when you talk about diet, you really talk about the microbes that live in us and around us, and more specifically also about the gut microbes. The gut microbes are of particular importance, and you surely know about that, because they have many beneficial aspects for us beyond short-chain fatty acids. They're producing precursors for neurotransmitters. They're producing neurotransmitters. They're producing vital amino acids. They help us to digest food ingredients that we cannot digest otherwise ourselves. And so when we get started with this work about 10 years ago, we really kind of realized we have to go to the gut microbiome. Now, as Jason nicely introduced the metabolic reconstruction approach, I will only glance over that. But traditionally, what has been done for the metabolic reconstructions is that you would take, obviously, the genome annotations, which and list all the metabolic enzymes and transports known to be encoded in a target organism that you want to reconstruct, as well as you perform an extensive literature review. And that meant that the PhD student spent many hours reading papers about the biochemical characterization of single enzymes for hundreds or even thousands of enzymes, and put that information together with the genome annotation in a structured format into the computer. Now, on the right-hand side here, you can see that what we collect at the end in the computer is really a knowledge base, because it captures what's known about the target organism in terms of metabolism and physiology based on literature. So really the literature review is crucial here. And hence this approach has been also called the ButMap approach. But at the same time, the way it's been encoded through the stoichiometric matrix that Jason introduced, it is also a computational model. And by using constraint-based reconstruction and flex balance analysis, we can actually very efficiently and quickly compute metabolic traits of these in silico organisms. And more particularly, it allows us to go large scale quite easily. And for many of these metabolic reconstructions, but by far not all, there exist also metabolic maps which help you in visualizing the content of the knowledge base and the computational models. So basically, these reconstructions serve as a blueprint for consequent condition-specific metabolic models. So one reconstruction can give rise to many different models which are constrained on experimental data, omics data, just as Jason showed you, but at the heart of it lies the metabolic reconstruction. And that integrates a biochemical, genomic, and physiological knowledge for the target organism. Now, here's shown the map for human metabolism, but there are for many microbes, them hand-drawn maps as well available. And so this visualizes you the content of the knowledge base. Each node here is a metabolite and each link is a biochemical transformation or a transport reaction. And so this wiring diagram is quite similar to a map of a city, if you wish, or a tubing map. And so the way the computations work here is that we can take a particular enzyme or gene product, map onto it experimental data, transcriptomic data, just as Jason mentioned, and tune how much flow or flux goes through every single of these links. And that's very similar to the pipe system or tube system shown here by opening or closing the valves. And with that, you change the properties of the metabolic model. And the beauty on the approach is that you can not only predict locally the consequences of such a change, but globally throughout the network. And this is, of course, also the case if you have different media composition or a different dietary composition. Think about a high-fat diet versus a high fiber diet. So you have different inputs. So you change the valves here, and that triggers throughout the network and enables you to produce different biomass yields or energy yields. Now, if you want to go to the gut microbiome, which is very personal, time-dependent, space-dependent, and different between the individuals, we're talking about hundreds of thousands different strains and species. So how do you accelerate this reconstruction approach that I showed you earlier? And that was a challenge we tackled a few years ago by creating a pipeline that is data-driven, of course, driven by the genome annotation, but also experimental data derived from the literature. And the pipeline that we developed makes great use out of the K-base, out of the Argonne National Laboratory, and also the Model Seed resource, which provides us with high-quality draft reconstructions. We found that particularly for gut microbes, which have less experimental data available and which might be phylogenetically quite distant to model organism, it is very important to have very good genome annotation. So we spend a lot of time and effort in the group to do comparative genomics using majorly PubSeed and refine the pathways and the gene annotations across the microbes that we are reconstructing. And then, of course, we use as much as possible biochemical data. We have an in-house database, and I will show you the link later on, which uses its own nomenclature, and for convenience, we translate the draft reconstructions into this in-house nomenclature. If there are novel pathways described for organisms in the literature, we tend to add those ones on a regular basis, and that will require us to extend the pipeline and make revisions to the pipeline. Overall, the pipeline looks like this. We take the draft reconstructions from either K-base or the seed, we convert it into our own nomenclature. We check for thermodynamic consistency. We apply a Western diet, which is basically a rich medium diet, to perform gap filling. And then we use information that we gathered from the comparative genomics to also further refine the reconstructions, correct the draft reconstructions if necessary, and also fill the gaps based on experimental data. And that is by now done largely automatically, but it does require to make certain adjustments and interventions manually and to inspect certain inconsistencies manually. So it's not a fully automated process, and to the best of our knowledge, it probably never will be, because there are always cases which are ambiguous and require more experimental data and additional review of literature. And then we have developed a test suite for quality control, both in terms of the ability of the reconstructed microbes to produce certain fermentation products, byproducts, or also grow on a defined media and carbon sources. And if the microbial reconstruction passes the test, we call it a semi-curated, semi-automatically curated reconstruction. If it doesn't pass it, it will go through another iteration. So by using such a semi-automated pipeline, we were able to largely accelerate the reconstruction process and generate, at the time it was 773 microbial reconstructions, which covered 14 different phyla. Now we have publicly available 818 of these microbes. And to our, well, those ones are accessible via the Human Metabolic Network database that I mentioned beforehand, and the link is given here. So you can pass it, or you can download the content, you can download the reconstructions, you can access the database via an API, or you can query its content via the website itself. Importantly, it also contains information about nutrition as well as human metabolism and disease. For us, quite exciting is there have been a growing number of papers using our resource in many different ways that we ever been able to think of. And that's quite exciting and rewarding because we do this work for the community and we share this work openly with the community. So it's great to see that researchers take on to that. Now, for our own interest, we got quite intrigued about really when we started working on data for Parkinson's disease is that many of the elderly people that are affected with Parkinson's disease do take additional drugs to the main treatment, which is levodopa. And so really the challenge for us became that how to account for drug metabolism, in particular there's increasing evidence that gut microbes can metabolize in one way or another on a large scale drugs that we take regularly. And so, so far, there are many reviews now available that describe in general the microbial activity, which can be either direct or indirect. And of course, drug delivery systems and pharmaceutical industry has been making use of these properties since many years by, for example, giving a prodrug rather than an active drug, which is then converted into a prodrug or an active drug by the gut microbiota. But also the microbiota can inactivate a drug or create toxic metabolites, which then might be in part at least causing the adverse effect that people are reporting. Then there's, of course, the antimicrobial and selective microbial properties of those drugs as well. But the indirect mechanism that also involves metabolism is that the gut microbes can produce certain metabolites such as paracresol that then interfere with host drug metabolism directly. So we set out to review the literature that's there about drug metabolizing enzymes and trying to represent them computationally. And so we found that to date, there are at least 15 known drug metabolizing enzymes, which are encoded by 25 microbial genes. Here's a table given. I'm just giving you the enzyme classes, the reaction types, and also the effect of the activation. Now, the important part is that many of this information has been generated using model organism again, and there is very limited knowledge available on a large scale of which microbes process which enzyme and has which activity. And there are very recently papers coming out now to describe on the microbiome level the drug metabolism, but there are very few papers, and still they do not characterize on the large scale per microbe the drug metabolizing capabilities. So using our comparative genomics approach, we set out for these 25 drug metabolizing genes to query over 5,000 genomes, gut microbiome genomes, for the presence and absence of these 25 genes, thereby trying to get a strain level characterization of drug metabolizing capabilities. And we integrated those ones then with the microbiome reconstructions. So we extended our first compendium that contained about 800 microbiome reconstructions to now more than 7,000 microbiome reconstructions. But we really focused our work on drug metabolism. And so we capture now these 25 drug metabolizing genes corresponding to 15 enzymes, their subcellular localization, which been predicted, and also we predicted 12 drug transporter genes. And that has been done across over 5,000 of these 7,000 genomes. And showing you the table from earlier on, again, we can now ask the question, well, what is the distribution of these different enzymes in the microbial strains, and which are the highest abundance ones? And what you can see is that certain central metabolic enzymes like the cytidine deaminase, which has, of course, other roles in metabolism than drug metabolism, is present in 80% of the microbes, while certain other enzymes like the dopamine dehydrogenase is only present in 3 of the analyzed genomes, consistent with other data that is out there. To really describe the metabolism of the drugs and enable actually drug-specific microbiome metabolism, we also went through the literature and described or identified the mechanism by which the microbes metabolize the drugs and the different drug derivatives. So we have done that for almost 100 different prescription drugs, providing now a very comprehensive database across many different drugs for many different gut microbes in their capability to metabolize those drugs. Here's an overview given in form of a circus plot that shows you which phyla can metabolize predominantly, or which contains predominantly which enzymes. And as expected and mentioned earlier on, the cytidine deaminase is found in 12 of the phyla, the nitroreductase in 13, and then there are specific drug metabolizing enzymes, which is consistent with the literature. Not so surprising, probably, the species with most drug metabolic capabilities were Escherichia coli species. There's a very Escherichia coli are very versatile, so that's not very surprising that they have many of these capabilities. We then went into the literature and identified information and collected information about microbes, either strains or species, and the ability to metabolize certain drugs. This information has not been used during the reconstruction or the comparative and genomics exercise. And so we got 238 drug microbe pairs. And the question here was then, if you take the metabolic models which contain drug metabolism for each of the microbes, how often do we predict correctly or incorrectly that they can indeed metabolize these the given drug? And you can find that we have almost 80% accuracy and a very reasonable sensitivity and specificity. So importantly, this is an independent data set. The reconstructions never seen these data. So I think that's quite motivating that indeed these reconstructions represent for drug metabolism what might be happening in the given organism. Now with this a large set of microbiome reconstructions at hand, we can now actually go further and ask the question, how do the drug metabolizing capabilities differ between individual microbes? And to address this question, we took a very comprehensive metagenomic data set from a Japanese cohort, actually, of 365 colorectal cancer patients and 251 healthy controls. Yoshida et al. Published that just last year. And we used our microbiome modeling toolbox, which is part of the COBA toolbox, to map the provided species abundance data for each of the samples onto the accou to reconstructions. The data were only provided on the level of species and not on strains, even though the accou reconstructions are on the strain level. So we generated, using the microbiome modeling toolbox, pan-species models. And in fact, we could actually map 97% of the named species, as opposed to some 70% if we were to use accou only. Now, we created a microbiome level model for each individual. So for each of these over 600 individuals, we have one model that represents the metabolism of each of the microbes that have been identified within the samples. We then subjected each of these microbiome models to an average Japanese diet that we derived from literature, and we added individually the different drug metabolites. And we asked, can we find or predict, using flex balance analysis, an individual-specific drug metabolizing potential, and does it for a given drug differ between individuals? And so here I'm giving you an overview of the drug metabolizing capabilities across these 600 samples and individuals. And you can see in blue where there is a conversion, just presence and absence, or qualitatively, if you want, in the microbes. And this is the case for most of the drugs. And in red, there's no conversion possible. And in fact, you can find the basalazide drug, which is the active form, the 5-aminosalicylic acid, and the inactive form is the N-acetyl version of this drug that only a subset of the samples that we analyzed can actually carry out the metabolism. And in fact, it's 42% of these microbes can do the metabolism qualitatively. Quantitatively, all of these microbes microbiomes show difference. Now, since those ones are mechanistic models, which basically have the metabolism of each species within a microbiome, we can actually go back to the models and ask the question, well, where are the bottlenecks? Which species are the bottlenecks for drug conversion capability? And actually which reactions are causative for that as well? And I'm showing you here the conversion of a basalazide, the activation from the prodrug into the active drug. And you can see that you had to have certain species in the microbiome in order to do the activation of the drug. And similarly, there are bottlenecks in the inactivation of the drug. And so this is one of the great advantages of the mechanistic model that permits us really to dig down into the pathway level and reaction level to identify what is happening and where are bottlenecks. Another example is levodopa metabolism and a digoxin metabolism. Both pathways are specific within the set of 7,000 microbes for Egatella species, and similarly, those ones provide the bottleneck. The first step, the conversion of levodopa into dopamine, is done by Enterococcus faecalis. And you can see here that actually there are differences between the individuals. So even though you have the enzyme present, not in all cases, you get a one-to-one secretion flux, predicted secretion flux for tyramine if you give levodopa with the diet. And similarly, this is not a one-to-one translation in the conversion of dopamine to tyramine down here, and that is to do with the presence of Enterococcus and Egatella and their relative abundance, as well as precursors. And again, you can go down to the pathway level and really investigate what's happening, what's missing, what could be substituted. You could start thinking about products and probiotics and prebiotics here to activate certain metabolic pathways present in the microbiome, or to substitute the lack of certain microbial pathways. We can also then, given the clinical data that were provided by the cohort, ask the question whether the drug metabolizing capacities vary, for example, with age or BMI or disease state. But we did not find any strong correlation with the disease state. We did find changes over the course of age for the ability to inactivate 5-aminosylic acid. And in fact, this ability increased with age. And similarly, there was a dependency of gemcitabine with BMI, whereby it increases the secretion potential with increasing BMI. So in summary, I presented you now a first-scale assessment of the drug metabolizing capabilities of the human gut microbes, which we done based on literature review as well as extensive comparative genomics. We have now a first time a large-scale description of strain-level capabilities of drug metabolism. We have defined the reactions carried out by the gut microbes for almost 100 different drugs, so that you can actually investigate the role of the different microbes, either individually or on the microbiome level, for these different drugs, individually or in combination. I showed you that we could use microbiome level predictions to distinguish quantitatively as well as qualitatively differences between individuals. And there it becomes very interesting to now starting to think about responders and non-responders who might develop adverse reaction to a particular drug and not, and what could be potentially a microbial cause for that. And how could one prevent that by either using an alternative drug or by administering probiotics and prebiotics. I demonstrated to you that we could identify limiting factors or limiting microbes and pathways in the different microbiomes, so bottlenecks, as well as correlate age and BMI, but we found also gender and disease state. We also investigated different diets in addition to the Japanese diets, and found that at least for some of the drugs, it's predicted to make a difference. Now, all that work would not have been possible with a lot of people involved. the driving person here being Almut Heinken, which is in my group, and she's been really the head of the microbiome work and driving the Agora 2. Dima Rafshi, which been driving the comparative genomics work, and many others have been involved over the years. I would like to help highlight that we have open position in the laboratory on all different levels, and I'd like to thank you for your attention, the organizers for giving me the opportunity to present our work, and yeah, I'm happy to take questions. Thank you very much.
Thank you, Ines, for such an amazing talk. And it was good to know like so much work has been done in your group for the microbiome analysis. I would like to thank both the speakers of the session today, Professor Jason Papin and Professor Ines Thiel, who have given such great talks during the session. And I would like to now open the Q&A session. And if the participants want to ask further questions, they can type their questions in the Q&A section here, which is down in your Zoom. So the first question, Jason, is for you. So Christian Diener has asked the question, like, what is a view on interaction between 3 or more strains? And are they more or less common than pairwise interactions?
Yeah, Christian, that's a good question. I'll confess that in our original experimental plans with the ASF, we actually wanted to do all interactions. So all pairs, all triplets, all 4 species. But then the student doing that begged that I relax that expectation because of the sheer amount of work that it would take. The answer to your question is I don't know. I would love to be able to do that. I expect that the interactions could be very complex. There's no reason to think that the interactions wouldn't be complex. It was just the sheer scope of doing those kinds of experiments that made it difficult. So I would love to be able to do that next though.
Thanks, Jason. The other question is, there's some data indicating that the spatial organization of the microbiome contains cluster rather than random distribution. So how do you think that would play into metabolite exchange across bacteria, or could the exchange be the basis for partners in these clusters?
Yeah, it's a great question. Yeah, the ASF community itself, actually, there is some spatial structure in the mouse gut for certain regions having a higher abundance of some species over the other. You know, I don't know that we looked directly to see if that spatial organization maps to the kinds of interactions that we observed experimentally. To the question, I think it's absolutely a possibility that the metabolic interplay and interdependence could influence that structure. Or the other way around, where maybe the structure for some other non-metabolic reason drives evolution of those metabolic interdependencies. So that's not something that we've explored, but I think could be a very important question to follow up on.
Thank you. Ines, the next question is for you from Huen Tran. Can you please share how you identify if the metabolites are microbial metabolites or host metabolites? Or can you distinguish like how these metabolites are microbial that are intracellular versus extracellular?
Okay, so generally this is done based on literature reviews. So in the parts of the drugs, it's being reported that a certain drug is metabolized by a microbe, and that's a conversion metabolism. And so that defines if it's a microbiome metabolite or not. As for if it is intracellular or extracellular, it's always a challenging question, particularly because the annotation of transport systems is and remains challenging. Again, if there's experimental data available, that's great. Much more should be done, particularly when you go to metabolites rather than to drugs. And then, of course, you can also use rules of thumb. regarding the molecular weight of metabolites.
Thank you, Ines. The next question is from Noah Rappaport to Ines. She asked, using your framework, could you go one step further and model the effect of dietary intervention applied to a personalized composition and combination with a drug metabolism capabilities, which will promote the individualized treatment success?
Great idea, and it's an obvious next step to do. It's a next step. So just need more hands, more people to work on it and make great, use our reconstructions that we provide.
Yeah, thanks. For Jason, how would you explain the comparably high number of amencillistic interactions on the pairwise level, and how does that transfer to community level interactions?
Yeah, from my understanding, amencillism is something that I don't know that there's clarity on what's the evolutionary role. So amencillism is where one of the pair of species has a growth deficit, so it grows less robustly, but then the other doesn't have a growth benefit. And we observed some of those in our pairings that we looked at. I think I would find it hard to believe that there's not some kind of evolutionary advantage or some kind of function associated with it, just because it seems to be, at least in our data, it showed up a few times. But I don't really have any understanding as to why, or what might be the benefit. Why might that explain, or could it explain some kind of spatial organization in the community or other kind of community dynamics? That's one reason why I think, back to an earlier question, I would love to do, and I hope that we can do soon, start to look at larger structures in that really controlled way. So looking at groups of 3 or 4 or 5 bacteria, because I think that will allow for addressing questions like this, where particular pairs might have some kind of interaction where there's a deficit, but maybe because of that, another bug can capitalize on that relationship. So that's something, I think, to answer the question, what I want to be able to do is look at structured interactions of a greater number of species. Some thoughts I've had on that.
That's true, yes. And also, I had a question about when you do the co-culturing experiments, so do you know if those organisms are phylogenetically distinct or they are very close to each other?
Yes, so that is known among the 8 species in the ASF community. Some are more related and some are pretty distinct.
Okay.
So there is a mixture of them. I do not believe we saw any correlation between phylogenetic relationship and the types of interactions that we observed. But of course, it's a very relatively small population size.
That's true.
Lord, so that would be an interesting thing to look at. Although I would also say that, in other work that we've done, just because 2 species are highly similar phylogenetically doesn't mean that there's necessarily a lot of overlap in metabolic functionality, or other kinds of features like that. So that's something to explore further for sure.
Thank you. For Ines, could you share your opinion about the advantage or disadvantage of the physiological effects of gut microbial metabolites in the metabolic syndrome based on the evidence obtained in clinical trials obtained so far?
I'm just rereading the...
Yeah, this is the question from Teresita.
Yeah. I'm not quite sure how to answer that. So there is, of course, line of evidence in the literature about the involvement of microbiome metabolites. And different groups have looked into, also computationally, looked into the roles of gut microbiome microbes in metabolic syndrome. We haven't done so. So I might not be the most knowledgeable person to comment on the metabolic syndrome per se.
Understandable, yeah.
But I can point out offline later on some great work by others that looked into that.
That would be great. Thank you, Ines. For Jason, so considering the RNA stability and other issues, how reliable is your experience, according to your experience, is the quantification of microbiome metatranscriptomic data, and what biases are typically found?
Yeah, it's a good question. It's not something I have a lot of specific experience with. My lab makes use of metatranscriptomic data. We're not regular generators of it.
Mm-hmm.
But I know it's a very active area of research to try to improve the quality of the metatranscriptomic data. I see that there's some related questions there about like proteomics. If I can just kind of fold that in, because it kind of connects to it.
Mm-hmm.
So is there a role for proteomics to validate that transcriptomic data? Just to go to Noah's question too. One thing I love about modeling as a discipline is it's not about, I would say is the ability to use the model as a framework to try to reconcile the data that you have. And so where the transcriptomics data that's integrated, maybe it's poorer quality, that doesn't necessarily mean that it's bad. It just means that you need to handle it with the model well. And so to the question about proteomics, that's a very active area of research with metabolic network models is how do we integrate disparate types of data? And can we use some data in a way, maybe we place a bias towards the quality of one data over the other. A challenge with proteomics, it's never nearly as comprehensive as transcriptomics data. And so I think that's why you see a bias towards gene expression, transcriptomics data integration. But there's certainly an opportunity to use both and in a way that allows it to confirm maybe transcriptomics data that's of lower quality. Mm-hmm. So modeling, I think one thing I love about it is the ability to try to reconcile and integrate to increase confidence associated with data, even when some of it may be of lower quality than you would desire.
That's
true. I think
true.
that was a really long-winded answer to that question. I just kept talking.
No, it's the true for both the metabolomics and transcriptomic data, since they are generated in a very different manner. So it's tough. Like there are so many methods available for integrating them with the models, but it's still like an ongoing research area. Yeah, and also like another question that is related to the culturing. So Moritz asked, have you tried to understand whether in monocultures or also in co-cultures, there's a separation of roles where within the same species, a part acts competitively and others cooperatively?
Yeah, Moritz, it's a great question. So not one that we've explored specifically, but I would say a couple of comments to it. So there's a great paper from Daniel Segre's group, actually, that I just was looking at recently, where he did this theoretical exercise looking at what kinds of interdependencies could be possible using metabolic network models. And so I'd point you to that for a little bit of that. But also to your question, Moritz, was something that we observed with the C. difficile example at the end. We didn't focus on looking at, is there a role for kind of cooperative metabolism among these phase variants, these variants in the C. difficile species, but there's clearly subsets of a culture for where the same species may take on one role that's interdependent with the role of another. So I think that happens probably a lot. How to characterize that becomes experimentally difficult. It's the same organism. And so maybe expression levels are different, but how do you map back to one sub population over another? That's something I think you could try to explore experimentally a little bit, maybe with that co-culture plate, but it's a great question. I don't have a good answer to, but.
Thanks, Jason. Moritz has more questions for Ines also. He asked about how do you check for thermodynamic consistency before applying a specific medium condition or diet?
Okay, so I had to look into my slides again, and I can see that is misleading the way it's been drawn. But of course, we have at the first instance a limited but yet not defined medium set, and we eliminate the obvious thermodynamic inconsistencies. Now, I think it's needless to say that none of the reconstructions go only once through the cycle that I showed, but it's many, many iterations. And hence, in the second iteration, you have the application of diet or medium constraints, and often we have a defined media if it's well available. So those ones can be then incorporated as well. So we do check with media conditions as well.
Thanks. And in your search for drug-metabolizing enzymes, did you also consider methods for predicting enzyme promiscuity and how that might expand involved genes or strains?
No, we haven't. And I think that goes along with another question, whether one can use the reconstructions now to predict novel genes and novel pathways. Mm-hmm. And functionalities. I think as these large-scale drug metabolizing characterization papers come out, like the two that I showed you, I think there's a great opportunity for the modeling to assist and help in identifying new candidates that might be able to carry out the metabolism. Either through existing genes, then you have the enzyme promiscuity, or through novel genes, because we couldn't find the corresponding genes in the genomes. Mm-hmm. So there's a discovery phase now, which is just exciting to be able to enter.
Thanks, Ines. Another question from Dr. Ali is, our experience with automatically reconstructed models such as agoras is that the predicted secretion rates of metabolites are often very high. So how cautious one should be when interpreting these results? Are they still qualitatively reliable?
So here I make a point again that we provide reconstructions. So a reconstruction is a starting point, and you incorporate condition-specific data. For example, the Western diet, and then you do predictions using the derived condition-specific models. So this is up to the user to integrate their own experimental data. We provide or can provide, upon request, simulations that we used in our papers, but often they're provided through the code itself. However, on the database and on the website, you find reconstructions. And if you take AGOA 1 or AGOA 2 and you constrain them accordingly, you actually will find that the secretion rates are often enough reasonable. However, we have no transport capacity rates. So they're the kinetic information that are missing. And this is really the next step to characterize those ones, to get Vmaxes, Kms, etc. incorporated to then do refined predictions to quantitatively accurately predict with these models.
Mm-hmm. Thanks, Ines. For Jason, the next question is, do you observe higher order interactions and how much do they contribute?
Yeah, we really need to do those experiments because this kind of questions come up a few times. If Rajita, if what you're referring to are higher order interactions, what you're talking about are like groups of 3, 4 multiple microbes. If that's what you're asking, then again, that's the experiment that we need to do. Or maybe you're referring, so I don't know exactly, if we were in person, I would ask you to clarify. If maybe there's a potential also, I guess, for looking at kind of stages or more dynamics of the interaction. You could envision where one microbe is secreting a metabolite that another consumes, that then causes or triggers that metabolite to secrete a new metabolite as a byproduct that then influences. So it'd be another kind of higher order interaction that I think could be possible. Importantly, with the experiments that we did, we grew it out until all of the bugs reached stationary phase. So a lot of the prep work was making sure we understood some of those dynamics. So we're kind of getting snapshots at the end of a growth period. Doing that with any finer time resolution would just be too difficult to do when we were doing it as a first pass. So that's how we focus now. So hopefully I answered two different interpretations of your question.
Thanks, Jason. I had another question. So the experiments that you showed in which the culture was growing differently, like the smooth colony and other colony.
Yeah, right.
So do you take into account signaling factors that would then like later affect metabolism in these cultures?
Yeah, so importantly, the images that I showed were done by a collaborator who in that paper in PLOS Biology explored a lot of the, or I should say some of the potential mechanisms associated with that phase, that phenotypic variation. So to your question, Priyanka, is signaling factors maybe playing a role in driving some of the metabolic differences? Very possible. This was a phenotype that was really only characterized with C. difficile just very recently. Our first question in that collaboration is, what role might metabolism be playing? And again, some preliminary modeling data suggests that there is interesting the shift between glucose metabolism and what looks like a Stickland fermentation metabolism, which as a shift within the same species driving different phenotypes is a really interesting discovery. And at least in some few days old preliminary data, it looks like that presence or absence of glucose could be something that's driving some of that variation that we're doing some repeating experiments now. So how much signaling might be playing a role in driving that, I don't know. I'm sure it's part of it, but that's something that's still being worked out.
Thank you. For Ines, like the data that you showed had around 100 drugs in them, like prescribed drugs. So were there any antibiotics in that list? Because antibiotics reduce the growth of the microbes.
Yeah.
And did any of these drugs reduce the metabolic capability of the microbes that you studied?
So we did not capture the antimicrobial activity of some of these drugs. So we only described the microbial activity on the drug. So what does a bug do to the drug rather than the drug to the bug? And this is deliberately done. Of course, one can always include in these microbiome levels simulations the treatment with certain antibiotics or the antibiotic features of certain drugs to reduce the growth rate of particular species or phylum when given to the drugs. But no, we did not consider the antimicrobial activity.
Great. Okay. Thank you. There are a few more questions directed for Ines from Tidre. I just wanted to clarify this Agora2 model relies on prior identification of documented enzymes, or is there a way to use this model to search for novel microbial enzymes with metabolic capabilities?
So we rely on documented enzymes. What this is a discovery phase that I mentioned beforehand, and which becomes really exciting. So you can try to think of in silico experiments that will permit you to hypothesize certain transformation activities. One of the things that we discovered is when we did the analysis of the independent data set, where we looked, did we agree or did we disagree with reported microbe-drug interactions. we actually had some 32 false predictions. So, well, we had more false predictions, but we had false negatives. And we went back into the genomes, and we found in, I think, a handful of these genomes that indeed the genes were present. We just had not annotated them. Across 5,000, I think that's a reasonable error rate. But then for the remaining ones, it's reported that they can do this microbiome transformation. Yet the genes, we can't find any autolog genes. So there is a potential there on the genomic part also to drive now further discovery, try to search the genomes for potential candidates and then experimentally validate indeed that they can carry out those activities. So I think that's a classical example on how you can use a metabolic reconstruction and their derived models to drive discovery.
Thank you for the detailed explanation, Ines. And the last question is from Dr. Ali. So are published manually curated metabolic models, such as those in big database, incorporated into Agora database?
Quick answer is no. There's a rationale behind it. The rationale is that when we started doing the gut microbiome reconstructions and the modeling with that we found the difficulty that when we use published reconstructions, our own that we had published in the past, and those ones published by others, particularly the Teusing group that had done lactobacillus reconstructions, etc. And we put them into community model or individually with a host, that we were never sure whether the organism could not carry out a particular biotransformation because it's not encoding for it, or because the microbiome reconstruction was never targeted towards a specific question that we wanted to ask, and that is host microbe interactions. For example, Bas's reconstructions were very much tailored towards yogurt production, which is fantastic, but he might have paid more attention to pathways that are involved in yogurt production while we are interested in host interactions. So there was also a bias in the reconstruction process, because at one point you just have to call a reconstruction finished. And I think you experienced that as well, Priyanka. It needs to be a finish line. And so what we then decided to do is to say we take a common bias, a common set of microbes, where we know it's biased towards host metabolism, towards gut metabolism, but they're consistently biased. Mm-hmm. And that is part of the reasons why, or major reason why the big database of reconstructions, which are very well curated and have, of course, a lot of experimental data incorporated, are not part of the compendium yet.
Thanks, Ines. The last question that came through one of the participants from Slack is it possible to analyze NMR data of gut metabolites from control and treated groups of animal in Agora and Microbiome studies, and how to analyze and find the correlation of metabolic, metabolomic, and 16S amplicon sequencing data if the treated and control group of animals?
Okay, so what we've done in the laboratory is that we used both metagenomic data as well as 16S data, and we predicted in the Agora reconstructions, and not MyCom, but the microbiome modeling toolbox, and predicted the metabolic secretion profile. So I showed you the drug secretion profile, but we can, across 400 odd metabolites, predict what would be the fecal secretion profile for different samples. And so then you can do a correlation analysis. More recently, and this is not yet published data or even bioarchive data, we were able to show that those predictions are actually or compare favorably with measured metabolomic data, quantitative metabolomic data, which is quite exciting if you could think about like that we could actually use computational modeling to predict what could be potential fecal metabolome, particularly since it's experimentally quite challenging to do a metabolomic analysis. So that we've done for human, but of course one can do the same for animals. Now, I'm not sure what Noah has for animals in mind. Agora 2 contains microbes found in mice as well, but there is a question which microbes are predominantly found in the animal of interest. But the methods are being developed by us and others, and these questions will be possible to address this.
Thank you both, Jason and Ines, for the amazing talk and such elaborate discussion that we had after your talk. I would like to, I'm very grateful for the time and effort that you have put in all the talk and share your work with the others. And yeah, we'll continue with the next session after 15 minutes. So thank you both, Jason and Ines, for agreeing to speak in the symposium and presenting such amazing work from your group.
Thanks.
Thanks, Priyanka.
Thank you.
Thanks, Priyanka. Thanks, everybody. Bye.
Bye.
Hi, everyone. I'm very pleased to open our next session of the symposium, which will focus on in vitro approaches to study the gut microbiome. This type of approaches mimic some aspects of the complex as well as complex to study gut environment and facilitate microbiome research. In this session, we will have two speakers, Dr. Thomas Gurry from the University of Geneva and Dr. Julie McDonald from Imperial College London. As in the previous section, time for Q&A will be right after the two talks. So please make sure to deposit your questions in the Zoom using the Q&A button at the bottom of the screen. And please note which of the speakers the question is intended for. You can also upvote other questions. Each talk will be 30 minutes. And after both talks, we will have about 30 minutes for questions to both speakers. Now, Thomas, is your cue to get ready while I introduce you. So it's my pleasure to introduce Dr. Thomas Gurry. Dr. Gurry completed his PhD work in protein biophysics with Colin Stultz at MIT. He went on to do his postdoctoral work with Eric Olm at MIT, studying the relationship between diet and the microbiome, as well as contributed to a number of clinical trials in different indications involving the microbiome. After his postdoc, Thomas focused on translational applications of the human gut microbiome, developing a combination of experimental and computational methods to engineer the fermentative output of the patient's microbiota in order to optimize clinical outcomes. Thomas is currently a senior research associate at the University of Geneva, as well as a co-founder and a CEO of Vesta Biosciences, which is a personalized nutrition company which was awarded a spin-off grant to develop a diagnostic for microbiome fermentation capabilities. Thomas will speak to us today about exploiting in vitro measurements of gut microbial fermentation capability towards disease prevention. Go ahead, Thomas.
Okay, thank you very much for the opportunity to talk. I'm just getting in full screen mode. Can you see my screen?
Yes.
Okay. Okay, so yeah, no, thank you. I really appreciate both that introduction and the opportunity to speak to you all today. As noted, I'll be discussing how we can exploit in vitro measurements of gut microbial fermentation capability towards disease prevention. The question that underlies much of what I'll be discussing today and the work that I've done in recent years is, can we engineer the metabolic output of an individual's gut microbiome through targeted dietary interventions, specifically in order to improve health outcomes? So one of the things that we started with here was to wonder whether we could modulate composition. And for this, you really have to go to an in vivo approach. And I would just like to contextualize why we take an in vitro approach by talking about this for a second. The issue is that actually, if you want to quantify the effects of dietary ingredients on microbiome's composition and even on its metabolic output, requires in vivo very tightly controlled feeding studies that are extremely difficult to pull off. Here is a study that we conducted at MIT, which was one where we were trying to understand the impact of different nutrients on the microbiome and its composition. And in order to do that and to try and mitigate the effect of variable diets and people consuming different portions of the prescribed diet, we actually put them on this nutritional meal replacement milkshake called Ensure that you may or may not be familiar with. And we enrolled about 60 people and asked them to consume nothing but that for 3 days, with water, of course, followed by a further 3 days of that with a particular nutrient spike in. And we collected stool throughout. The supplements that we were actually investigating were the dietary fibers, pectin, inulin, and cellulose, as well as coconut oil as a sort of fat, saturated fat, fish oil, unsaturated fats, and protein powder. And what we found is that we didn't actually see any signals from the non-fiber nutrients. Perhaps that was the study was underpowered, but certainly we saw some reproducible effects from dietary fibers in vivo. So here are shown time series with the squares corresponding to the days in the figure above, and in blue is the relative abundance of that particular bacterium is shown in blue if lower than average for that particular trajectory, and in red if higher. And you can see that these bacteria were found to statistically significantly spike on days 5, 6, and maybe the first day after the intervention, which corresponds to exactly what you would expect from a bacterium responding to the spike in specifically. And in particular, we were seeing pretty strong responses from inulin and pectin. So indeed, we can see some reproducible effects. However, we also see some individual specific effects. To illustrate that, on the left-hand side, I'm showing a case where it's a fairly homogeneous response across participants. You can see that particular Bacteroides uniformis species has a relative abundance that blooms on days 4, 5, 6. In the arm where patients were treated with inulin, whereas in other arms there's no significant bloom. And when you break that down by participant in the lower panel, lower left, normalizing their relative abundances to day 3 so that they're comparable, you can see that all of them are blooming to a certain extent and multiple factors of the original abundance. However, on the right-hand side, another Bacteroides species, Bacteroides cellulosilyticus, which you may or may not have heard of and is a cellulose degrader. If you look at the average time series, you may get the sense that cellulose is resulting in reproducible blooms. But actually, if you tease out the individuals, there are blooms in the different participants of that arm. But there's one individual in particular here, shown as AO89, that results in a 30-fold increase in the relative abundance of that bacterium. So there seems to be something about that individual's gut ecology on the one hand, potentially, or a strain specificity of that particular individual that results in their microbiome having a culprit that's very, very capable of feeding off cellulose and fermenting it. The other thing that I will note that complicates matters and really does lend itself to in vitro investigations where we can probe more carefully is that the ability to ferment a given fiber is likely strain specific. Again, honing in on the Bacteroides uniformis example where I'd shown it with the response to inulin. What we did is that we went and calculated from the single nucleotide polymorphisms in housekeeping genes of that species. We calculated the heterozygosity for a given individual within that individual. And loosely speaking, that's a proxy for the strain level diversity, if you like, how many different, what are the fractions of the different polymorphisms that you see at that site. And so high heterozygosity is a proxy for high strain diversity, if you like. What we see clearly is that here shown at 90, 97, and 99% identity to the housekeeping genes, just a different alignment stringency, if you like. You see that on day 6, there's a reduction in the heterozygosity within individuals, which indicates that there's a lower diversity in that species, despite a higher relative abundance, indicating that a particular strain is blooming. And when you look at the between subject heterozygosity, you can see, again, particularly at higher alignment stringencies, that the heterozygosity between subjects goes down on day 6 as well, which indicates that not only is there a particular strain responding, but it appears to be the same strain across different individuals. Now, this is merely suggestive, but I think that this is consistent with what people know about dietary fiber. And then, so why are we, really curious about dietary fiber? At the end of the day, in order to understand the motivation here, I mean, there are over 1 billion people currently on the planet suffering from chronic conditions, either of inflammation or metabolism. And one of the key processes in the background that's happening in all of our guts right now is the fermentation of dietary fibers by the gut microbiome, which results in the production of short-chain fatty acids, for the most part acetate, propionate, and butyrate, as you'll probably know very well. And these molecules have wide-ranging impacts on host physiology, which have downstream effects on processes and biomarkers that are very likely associated with different diseases. Some links are stronger than others. For example, IBD and diabetes, the link is quite strong. And in others, they're merely suggestive at this point, but there's mounting evidence. So really, this is the sort of context behind which that causes us to want to understand fermentation deeply. And I'll just note, as I said, that SCFAs impact host physiology in a variety of ways, and through two main mechanisms. The first being by binding G protein coupled receptors in the gut epithelium on the one hand, and in immune cells on the other, and potentially other tissues as well. But the other is through HDAC inhibition. And to cut a long story short, those of you who aren't familiar with that, all you need to know is that HDAC inhibition results in different epigenetic regulation. And so actually short-chain fatty acids can impact host tissue gene expression. And there's no real reason why that would be limited to the gut, since these short-chain fatty acids are found in the blood and circulating. And so there are many potential ways in which these can affect the host. The second thing that I would just note about short-chain fatty acids, particularly in the context of our symposium today, is that they're the output of very complicated ecological networks in the gut. And so we've very much taken a black box approach here, where we've black boxed the microbiome and consider fiber in, short-chain fatty acids out, and that complex ecology as very much inside that black box. But it's really important to note that there is that complexity which complicates analysis. And so the last thing that motivates an in vitro framework is that measuring SCFA production in vivo is very difficult. On the one hand, we don't have any technology that allows us to non-invasively track fermentation in the gut. And this is obviously because this is happening inside the gut and as a continuous process. But also, something very important that I think sometimes people tend to neglect is that stool short-chain fatty acid concentration, while interesting and while resulting in signals in associations with disease, may not necessarily be so representative of colonic SCFA production. And the reason why I say not necessarily is that it may or it may not be as a function of different parameters. So for example, just to illustrate this, if you make a very simple model of short-chain fatty acid production where you have fiber ingested and fiber excreted, and the short-chain fatty acids are produced at a certain rate as a function of the microbiome, here depicted by X. And then they are, they are both degraded and absorbed. Degradation doesn't really happen in a meaningful, quantitatively meaningful way on host timescales, but however, absorption is a big part of it. And all indications from the literature are that in most concentration ranges that are physiologically relevant, that absorption is linearly dependent on the concentration. So you can construct a very simple model here where you have a rate constant, absorption rate constant, and depending on the value of that rate constant, which we currently don't know accurately, the interpretation of stool SCFAs is very different. So for example, here plotting butyrate on the y-axis and different values of that rate constant on the x-axis on a logarithmic scale, plotting the total butyrate excreted in blue versus total absorbed in red, depending on your value of that rate constant, you get a very different interpretation. And so one estimate in the literature was obtained from a dialysis bag approach. Another was using a gut-on-a-chip monolayer, which we conducted preliminary experiments with, and we obtained a rate constant that was almost 2 orders of magnitude off of the dialysis bag approach. We don't claim that ours is any better. All I'm saying is that the dialysis bag rate constant results in an interpretation where the short-chain fatty acids excreted are basically the entire amount of short-chain fatty acids produced and only a very small fraction are absorbed, versus in the monolayer rate constant, the interpretation is quite different, and there's only a fraction of it that's actually excreted. And so you have to ask yourself, was all the substrate fermented before the stool was passed? And a lot of these different questions that might actually completely alter your interpretation of the results. This is why we've resorted to an in vitro, or put differently, an ex vivo framework, where we collect fresh stool from volunteers, homogenize it into a slurry. It's an extremely simple framework where we incubate it anaerobically at 37 degrees and spike in different conditions, either a control condition with nothing or different dietary fibers, as in the case here. And through time, we can sample aliquots and measure short-chain fatty acids. So what we find is that fermentation capabilities differ quite dramatically between people. On the top, I'm plotting here butyrate in both plots, butyrate concentration as a function of time over a 24-hour time series. And different conditions, namely different spike-ins, are plotted in different colors. You can see in this particular subject that they're very good at making butyrate from inulin, but they can't measurably make butyrate from pectin or cellulose above the control condition. In contrast to the second subject, where they seem to be able to make it from inulin and a bit less from pectin and a tiny bit from cellulose. What you can do then is that you can measure somebody's fermentation capability, if you like, which we define as their ability to produce a particular short-chain fatty acid from a particular fiber and across the pairs of fibers and SCFAs. And so that's perhaps what one subject's fermentation capability would look like if you limit yourself to inulin and pectin. And you can repeat this over many people, which we did. And what you find is that people do differ, as you can see, in their fermentation capability. And moreover, they tend to cluster into what appear to be discernible groups. That may be an artifact of clustering. At the end of the day, it's a continuum. But nonetheless, it's interesting to note that, for example, type 2 are very good at making all 3 short-chain fatty acids, but from pectin, as opposed to, say, type 5, which appears to be weaker than the population average in general at making all short-chain fatty acids from those 2 fibers. And so these different people have radically different fermentation capability. I'll also note that fermentation is highly specific to fiber type. So we extended these experiments to a larger set of fibers. And as you can see, the results are similar. Here on the left, showing propionate production rate computed from the early linear phase of the production on the previous plots. And you can see, for example, that this individual varies by fiber in their propionate production rate, similarly by butyrate, in butyrate, and it really does depend by fiber, and that is true the more you extend the list of fibers. But the thing to note here is that people often talk about soluble fibers as being the same thing when they're being sloppy as fermentable fibers, and insoluble fibers as not being fermentable. And that's, strictly speaking, not very accurate, because depending on the model that you use, you may not find that. Here we find, for example, that microchitosan is well fermented into both propionate and butyrate by this individual, which is an insoluble fiber. Similarly, the degree of polymerization appears to be a very important variable. So, for example, both inulin and fructooligosaccharides, of FOS, are derived from chicory root, if you like, or as one example, and they differ only in the degree of polymerization. They're both fructans, and FOS are just a shorter chain length, typically, on average. But you can see that in this individual, as far as propionate production is concerned, they can't make any propionate from inulin, the long form, but they can make a lot, as much as anything, from FOS. And all that's changed is the degree of polymerization. Similarly, if you take chitosan and microchitosan, both chitin-based fibers that are modified chitin with different degrees of polymerization, you see the same effect both with propionate and butyrate, that microchitosan is well fermented, whereas chitosan is not fermented at all. So one has to really be quite precise if you want to be talking about the ability to ferment a given fiber and one should be careful to clearly define the degree of polymerization and everything. And frankly, the field hasn't gotten to that stage yet, which is something that I think we all need to graduate to, me included. But we can use such a framework to also measure fermentation of proteins and amino acids using the same method. So for those of you who are perhaps less familiar with that, what tends to happen is that there are branched chain amino acids, specifically leucine, isoleucine, and valine, that are fermented by the gut microbiome if they make it down there. So if you ate it in excess or at a very large quantity and they make it down there to the colon, they're fermented into their equivalent branched chain fatty acid, which are a type of SCFA, if you like. But so, for example, valine is fermented into isobutyrate, and you can see that as you dose valine into the medium, you see an increase in the amount of isobutyrate produced. Similarly, if you spike in leucine and dose it from 1 gram to 10 grams, let's say, you can see an increase in the amount of isovalerate used. So it appears that this in vitro framework, while it may not be perfectly representative of the gut, and it is a very simplistic model, I do recognize that, allows us to probe the fermentation of different dietary ingredients. And one thing that I think I'll note as well is that microbiome composition can predict fermentation capability. It's not so surprising, since at the end of the day, the bacteria are the culprits. But here I'm illustrating this in terms of short chain fatty acid and fiber pairs, propionate being on the top row, butyrate on the bottom row, inulin on the left-hand column, and pectin on the right-hand column. And you can see that on the x-axis is the relative abundance of this particular lactose Boraciae OTU that's unknown, of an unknown taxonomy. You can see that the relative abundance of that OTU is correlated with the ability to produce butyrate from inulin, but less so with other pairs. However, in contrast, you have, say, this Prevotella copri OTU, where there's quite a clear correlation between the ability of to produce propionate from inulin, but not much else. And that would be consistent with what we know about Prevotella copri, namely that it's a propionate producer, like all Prevotella. But so the last thing that's important to note in terms of is this actionable information, or is this just a snapshot that changes in time entirely? What we did is that we re-recruited the same participants several months, up to 6 months down the line, and then compared their fermentation capability between time points. And here on the left, you can see them at both time points. They do differ between individuals, but the extremes tend to be preserved. And to illustrate that, if you project anything lower than average, or blue, onto, say, low, and in blue here, and anything higher than average onto high, and binarize, if you like, your data, and you do a two-tailed Fisher test on the similarity between those, you can see a statistical significance. So there's indication that fermentation capability, at least in its extremes, is relatively stable through time, of course, assuming that, and this was an exclusion criterion for us, you weren't treated with antibiotics, you didn't undergo major lifestyle changes, and so on and so forth. But that is somewhat actionable, because if there's some stability over 6 months, then we could presumably lead to a clinical vision for a particular ICD-9 code, for example, where you would have a group of patients that could be stratified according to their microbiome fermentation capability, and assigned to different personalized dietary fiber mixes as a function of their fermentation capability. Some patients may require more than that if their fermentation capability is so impoverished that they need some kind of recolonization therapy. But ultimately, if you can link the production of a particular short-chain fatty acid or other fermentation metabolite with a particular ICD-9 code, then this diagnostic framework could be somewhat helpful. And what it also allows us to do, this in vitro framework, is to measure fermentation capability in different patient groups. So that allows us, in a sense, to do case control studies between diseased or healthy, or different different stratifications, We're doing that currently in a cohort of Alzheimer's disease patients, and specifically around particular mutations. And as well, since as some of you may know, there's a link between dietary fiber intake in response to cancer immunotherapy, we can look at the difference between responders and non-responders, and we're also exploring that at the moment. But at the end of the day, if we want this to be clinically actionable and translatable, we need to be able to measure the impact of fiber supplementation on the host. And before we can even get to that, we need to be able to have a good readout of the process itself. As explained, we don't have a good readout, and that's why we resorted to the in vitro framework. But if we want to go back to the clinic, we need to be able to do something. And so the question that we're asking is, can we use plasma short-chain fatty acids as an in vivo readout? And also, can we identify high-quality surrogate biomarkers of microbial fermentation? For example, circulating markers of inflammation, such as pro or anti-inflammatory cytokines, or potentially other metabolites as well. And plasma short-chain fatty acids are very interesting to me. I find that they're understudied because, as I noted, histone deacetylase inhibition, or HDAC inhibition, is something that can transcend the gut, and in theory. And but the problem is that short-chain fatty acids exist in plasma potentially at sub-micromolar concentrations in some people. And depending on the literature that you read, the estimates vary by 2 orders of magnitude. So we need to be able to measure these very accurately and sensitively, much more so than from stool, by 2-3 orders of magnitude lower concentration. And so to conduct these analyses, we've developed an in-house LCMS protocol with internal standards specific to each short-chain fatty acid and with a very low limit of quantification in the nanomolar. And so we went to some length to do that, which is now allowing us to accurately quantify plasma short-chain fatty acids in order to study their pharmacokinetics, which is something that's poorly understood. And so the study design that we're taking here and that we're currently ongoing, I wish that we had data to share today, but that'll have to wait a few months, unfortunately, is to treat a participant with different meals. For example, in our study, a high fiber diet in the first meal, a low fiber diet in the second meal, and then a third meal consisting of the same low fiber meal as meal B, but with a particular fiber chosen to boost butyrate in this case, and personalized, determined using our in vitro framework. And then we can track both the short-chain fatty acids as well as the surrogate biomarkers. So with that, I'd just like to thank my collaborators and as well as Professor Scaposa, with whom I work at the University of Geneva, and my postdoctoral advisor, Eric Alm, with whom I started all of this work back in the day, as well as Dr. Tu Nguyen, who was a close collaborator of mine as well, and with whom we started that pilot experiment and developed this in vitro framework. So I'm very grateful to her for that, as well as Professor Frizzoni, with whom we're currently doing a lot of these analyses, including the latest diet study. So with that, I'd very happily answer any questions in the next session. And I do very much appreciate the opportunity once again. So thank you very much.
Thank you very much, Thomas, for this fascinating and clearly explained talk. Without delay, let's move to our second speakers. Dr. Julie McDonald from Imperial College London. I'd like to remind you to deposit your questions using Zoom in the Q&A window and state to which of the speakers the question is intended. Dr. Julie, this is your cue. Dr. Julie McDonald completed her PhD in the lab of Dr. Ellen Vercoe, where she developed an artificial gut model to study the human gut microbiota. During her postdoctoral research, she moved from Canada to the UK to pursue training in metataxonomics and metabonomic techniques. After her postdoc, Dr. McDonald joined the Imperial College London as a lecturer to continue studying microbiomics and metabolomics. The research in our group focuses on investigating novel methods to reestablish microbiota-mediated colonization, resistance in patients with intestinal pathogen colonization. Her lab uses a variety of complementary approaches to study gut colonization resistance, including artificial gut models, aka chemostat or robogut models, batch culture experiments, patient fecal samples, and mouse intervention experiments. Dr. McDonald will talk to us today about using artificial gut models to study how the gut microbiota protects against intestinal infections.
Thank you very much. I'm really excited to be here today. All right, so research in my lab has been focusing on investigating gut microbiota-mediated colonization resistance. What I'm referring to when I talk to that is how the gut microbiota protects us from infection or colonization with pathogens. So things like Thomas mentioned in his talk, short-chain fatty acids and production of bile acids. These are both groups of metabolites produced by the gut microbiota. Nutrient competition, so can members of the gut microbiota outcompete pathogens for these essential nutrients? Are members of the gut microbiota producing bacteriocins? And also phage abundance in the gut microbiota. So some phage can specifically target mice pathogens. There's also things like the mucus layer, which is a more indirect method of colonization resistance that's also modulated by the microbiota. So just sort of a broad overview of a lot of the different techniques used, or sorry, a lot of the different components of colonization resistance. But what my research is looking at is primarily at the moment focusing on metabolites produced by the gut microbiota. And one aspect of the gut microbiota I've been looking at is this idea of fecal microbiota transplantation. So this is where we take feces from a healthy volunteer and we homogenize it, and we can then prepare it usually as a liquid slurry, but sometimes as a capsule form. And we can give it to a patient that's ill. An example of this being something I'll talk about today, Clostridioides difficile infection, or you might also know it as Clostridium difficile infection or C. diff infection. But the idea here is you're trying to administer a healthy microbiota to try to restore the microbial ecology of the gut. And you can administer this preparation via two different routes, the north or the south route, so by nasoduodenal tube or by colonoscopy. So this idea of fecal microbiota transplantation is considered to be a first generation microbiome therapeutic. So again, we're trying to replace the entire ecosystem of the gut. But compared to other different drugs, it's got a relatively unknown composition. So even if we sequence the sample fully, you're still going to have unclassified members of the gut microbiota, and these members can change in abundance over time. Unless you're screening each and every single stool sample and quarantining it until you have the results of that screening, you're potentially have this risk of transmitting infections. So potentially sometime between the time the stool was donated for the screening process and when the donor donates the sample that's going to be used in the FMT preparation, you're potentially going to have a window where they could pick up an infection. Or even sometimes you have the case where certain donors are not screened for specific infections, or perhaps the screening tools aren't sensitive enough to pick up specific infections. So you have still have that risk of transmitting a pathogen using FMT. Also with this, you're relying on engraftment of bacteria within the gut. So this can in part be dependent upon the recipient's gut microbiome diversity and perhaps when they were last on antibiotics. And if they receive antibiotics after their FMT treatment, that's potentially going to undo what you were trying to do with FMT in the first place. And that's not completely unexpected, especially if you're dealing with groups of immunocompromised patients where they do tend to be put on a lot of antibiotics. We don't yet have a great understanding of the long-term effects of FMT as well. So if we think about things like if we think about diseases that take a long time to develop, maybe things like colorectal cancer, we know the microbiome can be involved in colorectal cancer, but we don't yet know if perhaps administering an FMT from a donor now, for example, may or may not contribute to development of colorectal cancer in a recipient, say 10, 20, 30, 40 years down the line. And finally, sometimes with specific diseases, we seem to think that certain donors could potentially work better than others. And we don't really know a good way of identifying these donors until we really try them. And the other thing is you risk potentially losing that donor if they have to go on themselves to receive antibiotics, or if they have some sort of big dietary change or lifestyle change. So this is what led to the development of second-generation microbiome therapeutics. And the idea here is that instead of using an undefined fecal preparation, we're trying to specifically isolate bacteria and give them in a defined mixture. So grow as many bacteria as we can from stool, and then choose specific bacteria that we've grown in pure culture and mixing them all together to make this synthetic cocktail of bacteria. But if we're going to use these therapies to treat diseases, we need to have a good idea of what bacteria we want to include. We need to ensure that they're safe and they're not going to contribute to other diseases. So it seems like almost every week we see, bacteria X is correlated with disease Y. So we really need to make sure that what we're including is going to be potentially pathogenic. Again, we're relying on these bacteria to engraft within the intestine to have an effect. And they could be, again, compromised if the patient goes on to receive antibiotics. But conversely, a lot of health agencies have required a sort of cocktail of antibiotics that could be administered to a patient if something goes wrong when they receive a synthetic cocktail. So they want the option to be able to remove the synthetic cocktail if there is something that goes wrong. But the more and more diverse these synthetic cocktails can be, the more difficult it can be to try to find that cocktail of antibiotics. And to date, none of these live synthetic cocktail therapeutics have received regulatory approval. So still a very new field. So where my research is focusing on is looking at this idea of third-generation microbiome therapeutics, where we're moving away from using live bacteria and we're trying to look at specific functionalities of the gut microbiota that we could restore. So things like metabolites or microbial enzymes, something more well-defined that we can give the patient to try to restore their healthy state. So these are things that undergo more rigorous safety testing because we know exactly what we're giving them. We can administer it at a really defined and effective dose. So we're not relying on bacteria to engraft within the intestine, and sort of conversely, you can let the compound wash out of the patient more easily if there's a reason that we need to stop treatment. We can potentially deliver this via less invasive delivery routes. So ideally we could administer it as a pill or a liquid drink that would be less invasive than things like a colonoscopy to deliver an FMT. And we're not relying on donations from healthy donors, so we can ideally deliver it whenever we need to. So some research I want to talk to you today is some of my research looking at the mechanism of FMT to treat Clostridioides difficile infection or C. diff infection. If you're unfamiliar with it, infection with C. difficile occurs in patients when they receive a lot of antibiotics that reduce the diversity of their gut microbiota. And if they're then in an environment where they're ingesting spores of Clostridioides difficile, they then can germinate within the gut. So these spores can change from their inactive or more dormant state to their more active vegetative state, where they're actively growing and potentially producing these toxins that can cause disease symptoms, including things like diarrhea, pseudomembranous colitis, abdominal pain, and eventually even death. So ironically, the first line of treatment for C. diff infection is more antibiotics, and that can work to kill the vegetative cells. But if you have C. difficile spores that remain within the gut, once the antibiotics are stopped, these spores can then again germinate and cause this recurrent episode of infection here. And because the antibiotics cause this loss of diversity in the first place, it allows C. difficile to grow within the gut. That's where the idea of FMT came in, where we are administering a healthy fecal preparation to the patient to try to restore their gut microbiota diversity. And this previous study back in 2013 by Van Noot and colleagues showed that this is actually pretty successful. So if we look at a group of patients, we give them FMT, we can see that after one infusion of feces, about 81% of patients were cured. And if we then take those patients that weren't cured with the first infusion of feces and we give them a second infusion of feces from a different donor, we can see that that increases again to about 94%. And that's in comparison to the control arms of vancomycin treatment or vancomycin treatment with bowel of bladder. So you can see much lower rates of success. So actually this study was so successful that they had to stop it early because it wasn't considered ethical to keep assigning patients to these antibiotic arms here. So I just wanted to show you this sort of overview of what we know of recurrent C. difficile infection. So as I mentioned, antibiotic exposure and exposure to C. diff spores allows for vegetative growth of C. difficile within the gut. And that's what we call our first episode of C. difficile infection. Then we try to bring down that vegetative bacterial load by giving more antibiotics. But if those spores remain, again, they can bloom again once antibiotics treatment is stopped. But that would be your recurrent episode of C. difficile infection. And that's often where we consider giving FMT as a treatment. Usually when we give FMT as a treatment, we're also putting them again on more antibiotics. And then about 24 or 48 hours after antibiotics are stopped is when the FMT treatment is given. But there's still some questions regarding, as I mentioned, the mechanism of FMT, and that's what we were interested in. So what exactly is happening in the gut that is allowing this healthy state to be restored and is responsible for reducing the growth of C. difficile? So we had several hypothesis names of our study. The first was we thought that exposure to antibiotics kills bacteria that perform these important functions in the gut microbiota. We thought that the reduction or loss of specific microbial metabolic pathways created that environment that promoted C. difficile germination and growth. And we thought that FMT reversed the effects by restoring bacteria that were responsible for performing these key metabolic functions. So we aim to identify these metabolites and propose a new therapeutic, so this third generation therapeutic, to treat recurrent C. difficile infection, something that, as I mentioned, that is more well-defined, effective, and safe. So this is where I sort of come into this session of these talks here. A lot of my research looks at using these artificial gut models. As mentioned, another name for this is a chemostat or a robogut model. And I've got a little cartoon of one of those vessels here. So this is where we take a growth medium and we inoculate it with feces. And we're trying to culture the microbiota outside of the host. So this is the glass vessel shown here. And we have a growth media that's fed in at a specific rate, and it's been culture that is removed from the vessel at the same rate. And this is set such that the volume of the vessel is maintained and the transit time of the gut is mimicked. So we're pumping it at a rate that mimics the transit time of the distal gut. We're heating it to body temperature, and we're maintaining the pH of the distal gut. We're also bubbling through oxygen-free gas to try to maintain the anaerobic environment of the gut. And finally, we're just stirring it to make sure we don't have any sort of gradient buildup. This is a video of what this looks like here. Let's see if I can. All right, so hopefully this is easy to see. So this is an overview of just one of the units of an artificial gut model. So first, I'm going to start off by zooming in on that vessel that I just made a cartoon of right here. And this is where the gut microbiota is cultured. Move on to this part here. We have the gas service plate and the pump for the growth medium. This unit here controls the pH of the vessel. And this controls the pH and the temperature and the stirring speed of the vessel. And it's just the growth medium that we use to feed the artificial gut model. And people often think that's actually the fecal samples, but no, that's just the growth medium. Right. So this is what 2 units looks like. That's usually what we're running at a time. As you can see, they take a lot of space. So usually 2 units is what we run at once. And that way we can run one vessel as a test vessel and the other vessel is a control vessel. So if they're both inoculated with stool from the same donor, they're actually very highly reproducible. And this is nice because it allows you to really have that nice control for each of your test conditions. So this is just an overview of one of my studies, how we used FMT to treat C. difficile infection. And what we did was we performed 3 different experiments. And then each of these experiments, we inoculated 2 identical chemostat vessels with a fecal sample from the same donor. Then one vessel was treated with FMT and the other vessel was treated with saline as a control. We took a variety of different measurements over the course of the experiment. So this is a longitudinal study. So we have a baseline, an antibiotic treatment period, and then a FMT treatment period. We look for changes in the growth of C. difficile. We look for changes in metabolites produced by C. difficile. So I'm not going to talk about the bile acid data today, just focusing on the NMR data. We also look for changes in the microbial profiles using 16S sequencing. We then validated what we found in our in vitro model by analyzing fecal samples from C. difficile patients and from patients that were the FMT donors. And we performed these pure culture batch experiments where we incubated C. difficile with our compound of interest. And finally, we performed a mouse intervention study where we had a C. difficile mouse model of infection, and we administered our compound to see how effective it was as a treatment. This is just a brief overview of what we did in our artificial gut model experiments, so those in vitro experiments I mentioned earlier. So as I said, we homogenized our fecal sample, inoculated our vessels, allowed them to stabilize for 24 days. Then we dosed in some spores of C. difficile, and then dosed in a second dose of spores the next day, and began a 7-day treatment with an antibiotic that is known to make people susceptible to C. difficile infection. We then allowed that to stabilize again, and then we followed that by the administration of FMT to one vessel and saline to the second vessel. And then we followed the development of microbial community spot following this experimental treatment for a period of 12 days. So this plot here is showing you what happens to changes in the growth of C. difficile over the course of the experiment. So you can see in this little gray area here, so I should mention the y-axis is the CFUs per mil of C. difficile, and the x-axis is time. And this gray box here is showing you the antibiotic treatment period. So you can see with antibiotics, the growth of C. difficile increased in both of our vessels. And once antibiotics were stopped, we did see a slight decrease in growth of C. difficile, but it was still present in about 10 to 7 CFUs. When we administered FMT to the one vessel, we could see a drop in the growth of C. difficile, whereas when saline was administered to the other vessel, then it did not decrease. If anything, it increased a bit. So if we then compare the FMT treated vessel to the saline treated vessel, we can see that FMT caused a 94% reduction of C. difficile total viable counts compared to the saline treated vessel. And this plot here shows something similar, this time looking at C. difficile spore counts. So in this plot here, we saw an 86% reduction of C. difficile spores in the FMT treated vessel compared to the saline treated vessel. These plots here are something called string graphs, and it allows us to look at changes in the microbial composition over the course of these artificial gut experiments. So the thickness of the colored line represents the abundance of a bacteria, and the different colors are representing different taxa, where I've colored Bacteroides in blue, Firmicutes in green, Proteobacteria in orange, Verrucomicrobia in purple, and C. difficile is in red. On the right-hand side, we can see these are the saline treated vessels, and then these are the FMT treated vessels. And the little box here is outlining when the antibiotics were administered, and the vertical lines outlining when FMT is administered. And these are all weighted for bacterial biomass as well. So we can see that the antibiotics caused a dramatic shift in the composition of the microbial community, where we have a decrease in the bacterial biomass and a bloom of these antibiotic-resistant bacteria. After antibiotics were stopped, we do see some recovery of the microbiota, but C. difficile is still present in all of the vessels. And FMT treatment did cause a dramatic effect in the community, though it's hard to see with the color right here, but one easy example to see is that this donor was didn't have any Verrucomicrobia in their gut microbiota until after they received FMT. So as I mentioned, something we wanted to look at was changes in the microbial metabolites produced by the gut microbiota. We did this by NMR. And one metabolite which really stood out to us was a short-chain fatty acid called valerate. So I've drawn a little valerate molecule up here to show you what it looks like. If you're familiar with the other short-chain fatty acids, you can see this is one carbon longer than butyrate. So it's a C5 compound. And what we saw was before any antibiotics were given, we have valerate levels measured in all of our artificial gut vessels. And the concentration of valerate was actually positively correlated with the diversity. So you can see here, this is a more high diversity gut microbiome, and this is a more low diversity gut microbiome. So these are cultured fecal communities from a donor in his 40s, from a donor in his 60s, and from a donor in his 80s. In all three of these experiments, we found that antibiotics cause a sharp decrease in valerate levels. And in the vessels that received saline, those valerate levels remain low. But in the vessels that received FMT, we can see a restoration of the levels of valerate in all of our FMT-treated vessels. Another metabolite we were interested in was 5-aminovalerate. So here, it's a bit hard to see, but we generally have low levels of 5-aminovalerate. So the ones that has some a more elevated level for antibiotics was that low diversity donor. And then we can see antibiotic treatment caused an increase in the levels of 5-aminovalerate. And then in our FMT treatment caused a decrease in the levels of our 5-aminovalerate, and then the saline treated vessels still remained elevated. Another metabolite we're interested in was ethanol. So again, ethanol levels were generally low in the pre-antibiotic treatment period, apart from the lower diversity donor. Antibiotics elevated the levels of ethanol in all the vessels, and then in the vessels that were treated with saline, the ethanol levels remained high, and the vessels that were treated with the FMT had a sharp decrease in the levels of ethanol. And propionate is another metabolite we're interested in. It's a bit harder to see because they all had their own sort of set point or their own level of propionate that they were producing to begin with. But if we compare the test and control vessels, we can see that FMT treatment caused a decrease in the levels of propionate. And so if we start to piece together all these different metabolites, what was really interesting is actually we found that 5-aminovalerate is a precursor for valerate. And also the members of the microbiota can ferment ethanol and propionate to form valerate. So in other words, these compounds here are all precursors for valerate, and they all show the opposite pattern. So if valerate decreases, the levels of these precursors increase, which makes sense. So what about the other metabolites? I often get asked this question as well. There weren't really as many trends with FMT treatment in the way we designed our experiment. We can see things like butyrate had a bit of a decrease with antibiotics, as you'd expect, but by the time we administered FMT, those levels had already recovered. Same thing with acetate, isobutyrate, and isovalerate. One interesting thing we noticed was succinate was dramatically increased with antibiotics, and that's something that's been noted in the literature before. And what's interesting is that C. difficile can actually use succinate to support its growth, so it's something else that's creating a niche that allows C. difficile to grow. But actually, by the time we delivered FMT, the succinate levels had already recovered to baseline. So that's not responsible for the effectiveness of FMT, but it is something to note that might have helped C. difficile grow during the antibiotic treatment period. We then went on to perform correlations between some of these microbial metabolites and the members of the gut microbiota. And we can see here from this plot, we have, the way to interpret this plot is each of these little letters are different samples. And the closer together these two letters are, the more similar the samples, and the more far apart two letters are, the more dissimilar the samples. So you can see on this part of the plot here, we see separation between samples taken before antibiotic treatment, separated from samples taken during and after antibiotic treatment. So this is saying antibiotics cause a shift in the composition in the microbial metabolites produced by the gut microbiota, and these did not recover after antibiotics were stopped. We then go on to look at these correlation circle plots here. We can then sort of overlay this plot onto this plot here. So these samples taken before antibiotic treatment correspond to this part of the plot, and these samples taken after antibiotic treatment correspond to this side of the plot. And we have the different members of the microbiota colored in orange and the different metabolites colored here in blue. And the closer together the two words are to each other, the more positively they correlate. So you can see this here is Clostridium cluster 11, which is the genus that Clostridioides difficile belongs to. That's why I've outlined it. So basically C. difficile is corresponding with, positively correlating with succinate, which we would expect based on the literature. And we see a strong negative correlation here between valerate and Clostridium difficile, which is what we see in our experiment. If we perform the same sort of analysis, looking this time at during the FMT treatment period, we see a clear separation between FMT and saline treated cultures. And again, these same patterns where we see a strong negative correlation between Clostridium difficile and valerate. We then want to go on and validate our in vitro data by looking at fecal samples taken from C. difficile patients before and after FMT, as well as samples taken from healthy donors. And we measured the amount of valerate in their stool using a technique called gas chromatography mass spectrometry. And we can see that before FMT, valerate was completely absent from the stool of the C. difficile patients, in contrast to the healthy donors that did have valerate present. And then at 1, 4, and 12 weeks post-FMT, we saw that the levels of valerate were restored in the stool of the C. difficile patients. We then went on to perform some in vitro batch culture experiments where we took pure cultures of C. difficile and we incubated them with several different concentrations of valerate. And what we found was that valerate caused a direct decrease in the growth of C. difficile in a dose-dependent manner. So as you increase the concentration of valerate, you decrease the growth of C. difficile. And if we go on and look at some other gut commensals, we can see the same sort of thing. Sorry, we can see not the same sort of thing. We can see that members of the commensal gut microbiota are actually not as impacted by valerate compared to C. difficile. So Bacteroides uniformis was only had its growth decreased at the highest concentration. Bacteroides vulgatus actually had its growth promoted at the higher concentrations that we tested, and Clostridium sinens was unaffected by valerate. So we've since gone on and tested this with some other gut commensals and found that it does seem like valerate was affecting C. difficile more than other gut bacteria that we tested. And finally, we wanted to look at the effect of valerate in a mouse model of C. difficile infection to see if we administer valerate as a treatment, would it potentially work to decrease the growth of C. difficile? So what we did is we had two groups of mice. One group of mice received valerate in the form of glycerol trivalerate because we wanted to make sure that valerate wasn't absorbed too quickly. So we were orally gavaging the mice and we didn't want it to be absorbed in the upper GI tract or stomach. So this is what, again, reminding you what valerate looks like. And glycerol trivalerate is just basically 3 valerate molecules bound to a glycerol backbone. And the idea is that once it enters the gut and it experiences the host of microbial lipases, that's when these valerate molecules will be released and then would be more likely to be delivered into the gut. So again, we have our glycerol treated mice, and we have mice that receive PBS as a control. And then we measured the amount of C. difficile present in their stools over the course of our experiment. Now, what we found was that before we gave any intervention treatment, the levels of C. difficile were similar in both groups, or the exact same in both groups of mice. So that's what we want to see. And then after we started giving our treatment, we saw that valerate decreased the levels of C. difficile. So this is after just 1 dose of glycerol trivalerate, after 2 doses, and after the 3rd dose. So after the 3rd dose, we saw a 95% reduction of C. difficile growth in mice that received glycerol trivalerate compared to PBS treated mice. So just coming back here to this plot I showed you earlier, where we're trying to fill in some ideas of what the mechanism of FMT is to treat recurrent C. difficile infection. And now we can see that valerate levels are suppressed with antibiotics. They are not able to recover before the patient has to then go on to receive more antibiotics to treat their C. difficile infection. But once they're given FMT, the levels of valerate can be quickly restored, and that will allow us to suppress the growth of C. difficile. So that's just one pathogen I worked on. There's obviously many other bacteria that could potentially colonize the gut and cause disease. And that's where some of my research is going to be going, looking forward, trying to see other mechanisms that the gut microbiota can use to protect the host from intestinal colonization and subsequent infection. So I want to take a second to thank a lot of my collaborators and colleagues for their help with this study, as well as our funders. And I am happy to answer any questions. And thank you very much, everyone, for your time and for listening.
Thank you to both our speakers, Julie and Thomas. I'm very grateful for you for agreeing to speak and for your great presentations. We'll start the Q&A session now. So the first question is for Thomas. Have you looked at, it's from Cecilia, have you looked at whether providing pairs of other combinations of fibers results in different short-chain fatty acids production from each fiber in isolation?
Yes, we have. And but only in a very limited manner. It's an interesting question. And actually we're collecting data on that in a way to answer more rigorously. But it does appear that, for example, if you combine inulin and pectin in equal quantities, the resulting production, at least at the doses we consider, are a linear combination of the two. Now, that being said, that's at the dose that we're considering. And I think that we're potentially right in the sweet spot, you know, and there could be some saturation effects or some competition between pathways and so on. So it's a very interesting question, and actually quite a complex one when you get into the nitty-gritty.
Thank you. The next question is for Julie. is from Alex. Is in your artificial gut system, do you see C. diff bloom when antibiotics are not administered?
Yeah, no, it's actually not able to be maintained really without the antibiotic treatment. So under those conditions, the gut microbiota is doing, we now know a number of different things to suppress the growth of C. difficile. So it's producing valerate. Some data I didn't get to present, but other groups as well have shown that biometabolism is really important for C. difficile growth. So the gut microbiota also consumes a progerminant that's something called taurocolic acid. And so if you have a healthy gut microbiota, you're also consuming that, and you're also producing another inhibitory bile acid, so deoxycholate. So there's a lot of different ways that a healthy gut microbiota is not supporting the growth of C. difficile. So yeah, we need that antibiotic treatment.
The next question for you, Thomas, is actually a topic of a bunch of the questions that we've received is what drives the inter-individual variability in fiber fermentation capability, and is long-term dietary habits, early life exposures, or other factors altogether? And I'd like to connect it with a few other questions. You talked about that the fermentation capability is quite stable across time. So one of the other questions talked about how stable it is across age. And I was curious how stable it is across across diets. Have you tried to test it at a more finer time scale, like after a meal, after a different type of meal?
Right. I mean, it's a great question, and it's quite a rabbit hole, that one. I think that working back, we haven't tested long-term dietary interventions or even short-term ones, but it's a very interesting question. We do know, though, that if sampled between several days apart, we get very similar results. So that being said, when I say very similar, I say within the variability that you can expect between 2 different days of a stool microbiome. And so really, I think that the main drivers can be considered, at least at first order approximation, to be similar to the drivers of microbiome composition. It's a sort of functional property of the microbiome, the fermentation capability. And so diet does, of course, contribute to that. Long-term fiber intake will likely improve fermentation capability, just not through any selection pressure or other than simply providing the food for the bacteria that are good at fermenting it. So I think that in that respect, this is a rather simplistic answer, but the answer is yes, the diet does seem to be a big driver. And ultimately, I don't think that there's any higher order effects there that I can comment on at this point, namely perhaps some host feedback from, having production of certain metabolites that are of interest to the host, potentially immunity. That's something also, the effect of SCFA production on host immunity and the feedback back to the microbiome composition is something. These are things that are very poorly understood, at least by myself, and so I can't comment on. But I think that overall, the way I think about it is fairly lowbrow, and that it's the same thing that shapes your microbiome composition. So namely diet, lifestyle, environment, and so on.
Thank you. Next question is for Julie, from Namrata. Namrata is saying those are really cool results from valerate therapeutic in the CDI model. Is the benefit due to direct inhibition of C. diff, or is the valerate feeding the beneficial microbes and promoting their growth?
Yeah, so I did the pure batch culture experiments where I just took C. difficile and incubated with valerate. And I saw it did decrease its growth, but I did see that gut commensal also had its growth increased as well. So it could be a bit of both. So we do know it is by itself inhibiting it, but there could be other effects as well. Yeah, that's a good question.
Okay, the next question for you, Thomas. Your data from Namrata again, your data very nicely shows how specific stool samples might be biased towards production of certain short-chain fatty acids, even with the same fiber. But not all short-chain fatty acids are equal in terms of their benefits to the host. do you want your fiber intervention to remodel the microbiome to correct this bias, or else just capitalize on existing short chain fatty acid capability?
Well, I think that it's easier to do the latter. So as a first attempt, that's actually very much the angle we're taking, while a lot of people are working on microbial therapeutics consisting of actually trying to modulate the composition and so on. And that's fantastic. That is the future of the field. We're taking a sort of very low tech approach of working with what you currently have and saying, okay, if we can find a good enough links between short chain fatty acids and a particular indication, then we can say, try and boost butyrate production in a ulcerative colitis patient. And then we take that we just work with what they've got if they're capable of it. So ultimately, I think that rectifying but the very interesting thing, and this is something that we're collecting through time, and it's going to take some time, a couple of years probably, to get enough data on that. But through time, people taking specific fiber supplements and sampling their fermentation capability through time, we expect that just for the reasons that I was talking about earlier, if you eat the food for the bug is going to grow up and there'll be more and more of it, and likely then will reflect in your fermentation capability as an even stronger ability to produce that short chain fatty acid from that fiber. So I do expect that we can modulate and change that if that's what the question meant by bias. But certainly that's a sort of a more long-term goal that isn't yet found in evidence. I think what we certainly can do is take a particular patient, and assuming we can link it with short chain fatty acids, their indication, I mean, boost that production with their existing framework.
I have a question, might be a little funny, but to Julie, what do you think about, like, once you have transferred the poop donation into your platform and to your in vitro system, could you then do some form of like in vitro evolution of this sample to make it less and more resistant to, for example, CDI? And then could you then use this sample to put it back in the same patient and kind of alleviate or mitigate some of the risks for FMT?
As you say, like take someone's fecal sample, do something to it, and then give it back to them so that it helps them fight. I mean, that's a really interesting idea. I haven't really thought of that. I think, we had thought about taking a stool sample or a defined microbial community from our artificial gut system and then giving it to a patient, but not taking it from the patient to begin with. But I guess there are concerns. You need to make sure that you don't accidentally introduce contaminant, because obviously if you then give that to a patient, that could be bad. But so if you could very precisely control for that, that could be interesting. But then I guess if it gets back into the patient, you might lose whatever selective pressure you had that you were applying in your in vitro model. So you might have to maybe do other things like modify the diet or stuff like that to try to get those, whatever bug or population it might be, to remain increased. But it's interesting. I mean, I guess that's the nice thing about these in vitro models is you have a bit more freedom to do just kind of whatever you want. You don't have to worry about ethics, really. So.
Yeah, another related question, I guess, to you both is a lot of people are trying to actually change the composition, and like you both are focusing on like the metabolic capacity of the specific composition and how it can be changed. And you think like the future treatment would focus on like feeding or underfeeding certain species, or maybe is there a potential for these specific metabolites to actually be the treatment?
Julie, I don't know if you want to start. I'm happy to. I've got one thing I just want to say that's again fairly lowbrow, is that butyrate smells like vomit. Propionate smells like wet feet. So it's literally. I'm not joking. So it's sort of there is evidence, for example, in ulcerative colitis patients, that if you give them butyrate enemas, that you observe improvements in the disease course, but it's just not a treatment modality that's either patient friendly or even probably appropriate, given that we're dosing the short chain fatty acids extremely high, potentially at levels where we're impacting host physiology in ways we don't understand, as opposed to exploiting the chronic capability of the microbiome to produce it at healthy levels. And so In that respect, I tend to shy away from using the SCFAs themselves as treatment because we can exploit the bacteria to produce them in a chronic and slow release fashion. But I'll limit that to short chain fatty acids, and maybe that's where Julie might be able to, and short chain fatty acids, specifically butyrate and propionate and acetate. There are others, and maybe Julie might have something else to say about those, I don't know.
I'd add that valerate also smells horrible.
Yeah.
But actually, glycerol trivalerate hardly smells at all, which is interesting. So if you wanted to use glycerol trivalerate as a treatment, it is something that you can actually drink in theory. I've not tried it, but you at least can't smell it. But yeah, I mean, maybe if you're thinking about things like metabolites, it might be useful for the infection point of view. So maybe if you, for C. difficile, the problem is when you stop antibiotics, the C. difficile starts growing and you're not giving them a chance to recover their microbiota because they're just have to go back on antibiotics because they're sick again. But if you're able to suppress the growth of C. difficile and allow their microbiota to recover and use maybe valerate, for example, as sort of a temporary treatment that you're dosing in, and maybe once you stop that valerate treatment, once their microbiota has recovered, then you know what I mean? Like you give their own microbiota a chance to come back. So I think they can have applications, but I mean, I can see that if you're thinking of it from a dietary point of view, you're saying, okay, eat butyrate for the rest of your life. That might be more hard for some people, but maybe the companies might like that more because they can sell it. I don't know. But yeah, so I think it kind of depends on what you're looking at. But I think there's sort of different perspectives you can take on the idea of metabolites as therapies or bugs that produce metabolites therapies.
Okay, next question for Thomas is from Sean Gibbons. Are these fibers consumed uniformly across patients, independent of short-chain fatty acid production? Any evidence that gas, for example, CO2 production is higher in folks who are less efficient at producing short-chain fatty acids?
Mm-hmm. Yeah, I mean, it's a great question. We have limited data on that question, but in vivo, it seems that the only fiber that we tested in terms of it being consumed was inulin, and it was in a very accessible form and a particular degree of polymerization that made it such that we were almost barely detecting it in the stool after ingesting 10 grams a day. Now, when we look in vitro, we do see, I don't have the statistical strength behind our data to say this with full confidence, but that there seems to be a clear relationship between the produced total SCFA load and the amount of fiber fermented. And ultimately, those two are separated by some intermediates, but assuming those intermediates move along the chain, one converts into the other ultimately. So there should be a fairly direct link if you're able to ferment it. However, when it comes to the question of gas, so that's another really interesting and difficult question that I think no one knows for sure. However, I will say that all evidence points to the fact that producing gas is indicative of fermentation, which ultimately results in short-chain fatty acids. Also, the interesting thing is that the stoichiometry seems to suggest that different fibers will result in different amounts of gas. And indeed, we see that. It's actually very clear in vitro that inulin is a very gas-inducing fiber. I think people who've ever tried consuming inulin, at least a fraction of us will be able to say that indeed that's true. And indeed, that shows in vitro too. About 30% of people produce bubbles in vitro when you feed them with inulin. And that is related to actually a high production of total SCFAs in the medium. So all evidence seems to be that they're linked very much, but not directly necessarily, because it's specific to the fiber, I guess is what I mean. Pectin doesn't necessarily result in as much CO2 as inulin.
Thank you. Another question to Julie from Mario. Is there any information about the transcriptional response of CD to valerate?
No, not to my knowledge. So it's not something we've done yet, but could be something that we look at in the future. So I mean, I don't know anyone else who's looked at, I know some people who've looked at measuring valerate from stool from patients, but I don't know anyone else who's really looked at it sort of in a lab setting. But that's not something we've looked at the moment, no.
Thank you. Thomas, Sean is hitting again. Back to the diet question. I know that you enforced a similar dietary background across participants in your feeding study. Can you talk more about how that influenced the results?
Yeah, it introduced all sorts of artifacts that complicated our analysis instead of simplifying it. One problem is that the milkshake we used, Ensure, is very low in fiber, very high in processed sugars. Actually, it contains little else. It's mostly dextrin. So the result is that you're providing a very ubiquitous substrate to the microbiome. And what we saw, that's the hypothesis at least, is the ubiquity of the substrate, is that we saw greater stochasticity rather than in reduced variance between participants. In fact, some of that, if you recall, during the talk, I briefly talked about the strain level diversity in response to the diet. And what we found systematically was that strain diversity increased after 3 days of the standard diet, not only inside an individual, but between individuals. So it seemed to actually throw everything off have the opposite effect. And I think that was, rather than the choice of standardizing the diet, rather a poor choice of what we use to standardize diet. And indeed, that seems to be consistent with the fact that the signals that we find in feeding studies across the literature tend to come from fibers and ingredients like that. There are other signals too, but fibers are more reproducible than anything else, whereas feeding glucose to a medium is likely to introduce a bit of stochasticity. So that was one thing. The other thing I'll note is that huge amount of sugar, or I don't know what it was, but what we found was that the microbiome appeared to be quite stressed. We saw a great increase in the relative abundance of different phages, which were indicating that likely they were entering the lytic phase of their life cycle, and in response to stress. We also found much greater strain diversity across different organisms, and this across the whole tree of life, not phylogenetically constrained, which some evidence points towards the fact that the fitness landscape flattens when under stress, when a system's under stress. So overall, the effects did not seem to be good. And in hindsight, that was actually quite a mistake. But it was interesting nonetheless, and I think that people should stop giving Ensure in the clinic. That's all I'd say.
Thank you, Thomas. The two next questions I'll join together for you, Julie, is about the culture, the conditions of the culture. So the first question from Priyanka is the culture always in an exponential growth phase in the artificial gut setup, or have you observed the culture undergo dormancy? And then the second question is the culture medium provided at a more or less constant rate? And similarly, what is the rate of removal?
Good question. So the first question, I think they're generally considered to be in stationary phase. So not exponentially growing, but not dying. So yeah, stationary phase is the short answer for that. The next part of the question was about the addition of the media. What's the rate? Is that the question? Okay. So yeah, it's added it's constantly added throughout the entire experiment at the same rate. And the way it's set up is that once the level of the culture starts to rise, once basically it's a pressurized vessel. So once there's a little tube that kind of dips down like this, and once the level of the culture rises above that tube, it gets sucked out of the tube. So it's always maintained at that set level. So the rate of addition and the rate of expulsion are equal, so that you're not, because otherwise you're going to have the volume of the culture. Even if you have two pumps that are pumping at the same rate, it's not going to be the exact same, and you can have the total volume vary. So the way that we run them, the volume is constant and the nutrient addition rate is constant. And and that helps to maintain the stability of the community. So yeah, I hope that answers that question.
Certainly.
Yeah, I should have had a video for that, but yeah.
Thank you. So Thomas, another Tom, has a bit of an odd question. Do individual dietary preferences show any correlation with our ability to ferment specific fibers? In other words, do we tend to eat fibers that are more readily fermented by our unique gut microbiota?
God, that's odd but very cool question. I think that there's no evidence of that, but I love the question because there's plenty of reason to believe that we might over time, given that butyrate, for example, in large-scale cohorts where they try and correlate different metabolites with different lifestyle factors, butyrate seems to correlate most strongly with positive things and high quality of life scores in terms of just general wellness. And so it's only a step further to suggest that perhaps over time you would naturally, unconsciously be drawn to particular fibers that result in favorable short-chain fatty acid production. And so that could be some pretty interesting way by which these bacteria are like favoring their own selection. That's it's a high level effect that I think would be, I don't even know if it's that hard to test, to be honest. Maybe you could just, you just need very large cohorts with, high quality dietary data, which is a bit difficult, but diet questionnaires, well, actually, it's maybe not as difficult as regular dietary questionnaires because you could really constrain the question to fibers and go, okay, which types of fruits, vegetables, grains do you consume most? And gosh, I mean, that would be a fascinating result. But I think that's part of the feedback to our brains. I think that's pretty interesting. Yeah.
Thank you. Questions, Thomas. Is that okay?
It's the mash.
Okay. So years ago, I was talking to a colleague, and they were saying that they had a student who used to test their own microbiota in vitro. I believe it was with prebiotic of some sort. I can't remember which. They were saying that the prebiotic used to work really well. This person was single, lived alone, didn't eat great. And then they started dating someone and they started eating a healthier diet. And they found that the prebiotic didn't work as well because their gut microbiota in general was healthier. So I was wondering if you've ever come across anything like that with people changing their diets. And then as a sort of second question, I was wondering if you've ever looked at, because I'm interested, I'm kind of interested in antibiotics. I'm interested to look at somebody who's been on antibiotics. And then, so you have maybe a pre-antibiotic sample, a sample while they're on antibiotics, and maybe a sample while after they've recovered from antibiotics, and kind of see how their fermenting ability changes before and after. Maybe they haven't recovered fully, or maybe they have an alternative steady state. I don't know if you've ever come across anything like that.
No,
maybe you had a participant who accidentally went on antibiotics.
They're both great questions. I haven't looked at either of those, but I think that the antibiotics question would be very interesting to look at because actually I've tended to, especially when it comes to ethics committee protocols and things, just been like, oh, well, let's exclude, as everyone in the microbiome does, anyone who's taken antibiotics, let's get out of there. And the truth is that antibiotics, while especially broad spectrum are pretty damaging, that, there's really only a fraction of the microbiome that's responsible for fermenting these fibers. And it's unclear to what extent they're damaged by this. So I would be really interested to see. As I say, I assume that you take antibiotics that sit slate clean and then you're recolonized. But I don't think it's that simple.
No, well, at least in my in vitro models, you see some bacteria bloom. So in my case, I was looking at clindamycin, and there's lots of Bacteroides that are resistant, not all of them, but Bacteroides in general just have more antibiotic resistance mechanisms compared to some other bacteria. But yeah, I think people do consider it to be sterile, but you're not really.
Yeah, no, that's a really interesting question. I think this is something that we should actually actively study because ultimately antibiotics is something that we can't really do without, at least at the moment. And people take them and just saying, oh, well, you have to retest and nothing at all, no holds barred now. It's a bit cheap. I think there's some interesting science to be done there. So it's a great question.
Thank you, Julie. So another question to you, Julie, from media, and she's asking. She's first saying it was a great presentation, and you mentioned valerite suppressed C. diff infection. What valerite associate with? Does valerite associate with bile acids? Some bile acids known to induce C. diff infection. And just to note, from our talk yesterday, I don't know if you attended, but in our work, we have, together with Tom Wolmansky, we have noticed that bile acid had various different effects, differing association with gut microbiome diversity. Some of them were positively associated with it, some of them were negatively. So yeah, this question is about what you've experienced.
Yeah, so we looked also at bile acid. I didn't present the data because it would have been a really long talk, but so with bile acids, we have a better idea, I think, of a better idea of what members of the gut microbiota are involved in all the steps. We don't know exactly every bacteria, but we know some bacteria, lots of bacteroides, for example, produce this enzyme what's it called? Biosol hydrolase, space out. And we know some Clostridium species can produce 7-alpha dehydroxylase. These are some of the enzymes that convert, deconjugate primary bile acids and convert primary bile acids to secondary bile acids. And the conjugated primary bile acid torcolate does stimulate C. difficile growth and germination. But if you cleave off that taurine molecule, you get cholate, which is a more weak germinant. And then if you do the secondary bile acid synthesis from there, you get deoxycholate, which inhibits C. difficile. So in that sense, we have an idea. And that's something I worked on with someone named Benjamin Molish at Imperial College. And he did more work into detail on that with C. difficile. With valerate, people, as we've seen focus a lot on acetate propionate butyrate, which is great, but then we start to have questions, well, what bacteria are producing valerate? And there's some indications out there that valerate is associated with the high diversity of the microbiota. We think that some members, I think it's Silobacter, there's some member of the microbiota that has been suggested to produce valerate, but we haven't really gone through and teased out exactly which members of the gut microbiota are responsible for producing, and that's something that we will have to go through and do eventually, screen a lot of bacteria. But at the moment, that's not something that I'm working on currently. So this is the work I presented today was a lot of work from my postdoc, and I'm now starting my own lab. So I'm looking at a bit of a different idea of looking at the gut microbiota and antimicrobial resistance, but I thought I'd present my published work first. So yeah, so it's something that we're working on teasing out in terms of valerate.
Thank you. Thomas, as a question from Moritz Weber, have you considered testing a particular person's stool with your system to identify diet components that would favor stability or engulfment of particular probiotics believed to be beneficial to that person, maybe in a model-guided fashion in order to reduce the search space for metabolites?
Yeah, I mean, I had not considered that. That's a really interesting question. I think that what I would say is that we're not going to be able to necessarily say anything about engraftment. However, what you could say is that you could demonstrate that, say, if you have a particular probiotic and you identify which fibers are well fermented and used as a carbon source by that probiotic in vitro, then you can provide some evidence that you'll be able to increase its relative abundance transiently. Then when it comes to the mechanisms behind engraftment, that I'm much less qualified to discuss. And I think that ultimately that comes down to some other variables as well. But assuming a mass action effect, then I think that it's perfectly reasonable to say that, yeah, we could use that framework to at least increase our chances. I think that's a very interesting point.
Julie, a question from Sean. So FMT dislodges an established C. diff infection, but many people are colonized by C. diff and carry it asymptomatically. Have you explored colonization resistance in your chemostat system?
So like in a healthy gut microbiota? No, we haven't really looked into that too much because we were interested in looking at the effects of FMT, but at least from what we know about C. difficile and other pathogens, we wouldn't expect it to be growing at very high levels if it was present. And we don't consider C. difficile to be present at high levels, even in people who are asymptomatic carriers. Even in people who are on, we know have C. diff infection who are on antibiotics, C. difficile is not necessarily present at extremely high abundances. So it's not something not a lot of researchers I don't think have looked into it, because I think maybe it doesn't sound as exciting because it's not producing a disease. But I mean, I think another thing people forget about is that babies often have C. difficile in their guts, but they don't see disease in babies. But that's something that'd be interesting to look at, but not something I looked at personally.
Okay, we're at time. So I would like to conclude this Q&A session and thank both of our speakers for fascinating talks and a really insightful Q&A session. So we'll see you back at 12:45 with session number 3 that will focus on in vivo interventions. Thank you again.
Thank you very much.
Tomasz, can you hear me? Yeah.
I'm on mute. Yeah, I can hear you.
You can. Okay, I just wanted
can hear
to
you.
double check. Yep. No,
Yep,
sounds good.
everything
Okay.
sounds like it's set.
Wonderful. Thank you.
Hello, everyone. Welcome back from our short break. I hope that you're enjoying the symposium so far. My name is Thomas Wilmansky, and I'm a postdoctoral research fellow here at ISB, and I'll be moderating the last session of the day, session 3, in vivo interventions. We have two really fascinating speakers, Dr. Guaram Dantas and Dr. Joanna Lampe. As a reminder, please ask your questions via Zoom through the Q&A chat room. Each presentation will once again last 30 minutes, and it'll be followed by a joint 30-minute Q&A session afterwards. Dr. Dantas, do you mind sharing your screen with us? I'll provide a brief introduction.
How's that?
Yeah, I think that worked. Okay, great, thank you. So our first speaker of the session, Dr. Dantas, is a professor in the Department of Pathology and Immunology, the Department of Biomedical Engineering, the Department of Molecular Microbiology, and the Edison Family Center for Genome Sciences and Systems Biology at the Washington University in St. Louis School of Medicine. He received his PhD in biochemistry from the University of Washington under the guidance of Dr. David Baker, and his postdoctoral training in microbial genomics from Harvard Medical School under the guidance of Dr. George Church. Dr. Dantas' research interests and training lie at the interface of microbial genomics, synthetic biology, systems biology, and computational biology. His current research focuses on understanding and predicting how diverse microbiomes respond to chemical and biological perturbations, harnessing these insights to rationally design therapeutic strategies to curtail antibiotic-resistant pathogens and remedy pathological microbiome states, and engineering microbial catalysts to convert renewable biomass into value chemicals such as biofuels and pharmaceuticals. He is the recipient of several different awards, including the American Association for Advancement of Science Newcomb Cleveland Prize, the Harvard University Certificate for Distinction in Teaching, the NIH Director's New Innovator Award, and the Kenneth Rainin Foundation Breakthrough Award. Today's talk by Dr. Dantas is titled Predicting and Combating Pathogenic and Abiotic Disruptions to Diverse Microbiomes. Please join me in welcoming our first speaker, Dr. Dantas.
Great, thank you, Tomas, and thank you for the rest of the folks at the ISB for giving me this opportunity to tell you about some of the work in our lab looking at how sort of bugs and drugs interact with microbiomes. But before I get going, I think it's really important for me to acknowledge that I'm merely a spokesperson here. Anything of interest or importance that I might talk about today is thanks to the incredible group of people that I get to work with and represent. While I will highlight a couple of people who are responsible for a couple of the key vignettes, also please realize that all of those are team exercises, both with others in the lab as well as our many excellent collaborators. So the topic of my presentation today is this concept that bugs and drugs both alter the microbiome, but understanding those effects and predicting them requires an appreciation of context. And so we recognize that both drugs as well as bugs, so things like antibiotics and pathogens, can be destructive to the microbiome. But then there are also restorative potentials for so-called microbiome-directed medicines, things like fecal microbiota transplants, but also engineered or chosen probiotics. And it's really the question of how do we take into account the diversity and the composition, as well as features like diet, which I presume we'll hear in our second talk, and a variety of other environmental conditions that impact how those destructive or restorative properties manifest. And there's rich literature to show that both in this case, animal models show that dysbiotic guts in terms of low diversity microbiomes can enrich for pathogen infection. And then also in human studies, where here in a pair of papers, these authors showed that different microbiome compositions of individuals drives differences in probiotic colonization resistance. So let's start with antibiotics as one of these big features that can perturb the microbiome. We think of antibiotics as small molecules that target pathogens, but the reason antibiotics work is because they target all of these key cellular processes of bacterial life, which, as it turns out, are shared by both good and bad bugs. So effectively, as you ingest an antibiotic, you're hitting as many good bugs, if not more, as the infection itself. And indeed, through retrospective analysis, essentially at any part of the age spectrum in human life, there are a whole series of comorbidities associated with antibiotic use. The greatest comorbidity, of course, with antibiotic use is antibiotic resistance. And these numbers are probably familiar to all to know that antibiotic resistance encodes an incredible burden, both in terms of lives now as well as in the future, as well as economic costs. And unfortunately, exactly as resistant bugs are on the rise due to antibiotic use, unfortunately, antibiotics are not coming to market. And so with that context, we'd like to understand what heavy antibiotic use does in especially the context of developing microbiomes. So this first vignette is about our focus on the pediatric microbiota, specifically looking at preterm infants, acknowledging and recognizing that as medicine has improved in our ability to keep younger and younger kids alive, it also means that these kids are increasingly more vulnerable to infections and other pathologies because they're born too early. And one of those manifestations is in the microbiome. So we know that age-matched preterm infants versus term infants have a substantially lower diversity of species in their guts, sometimes as much as 10-fold lower during those first couple of months of life. And layering onto this, when we're thinking of antibiotics, it turns out exactly when this microbiome is getting set up, we bombard them. So we know that, especially very low birth weight infants get antibiotics, virtually all of them during the first couple of days of life, so especially if they're born about 10 weeks too early. And over the course of your stay in the neonatal ICU, where these kids are hospitalized, the top two drugs and 4 of the top 8 drugs that they receive are antibiotics. So in collaboration with Phil Tarr and Barb Warner at Washington University and St. Louis Children's Hospital, we've been studying for the last 10 years or so the impact of antibiotics in the developing microbiome. So in this first study, Molly Gibson, a graduate from our computational biology program, teamed up with Barb and Phil to look at a cohort of preterm infants who were hospitalized for their first 2.5 months or so of life. We had fecal samples available, and Molly stratified this cohort into a group of kids that only had antibiotics early in life versus a set of kids who had antibiotics early and then often. And through a combination of metagenomic interrogation techniques, she discovered that the bugs that dominate the guts of these kids due to antibiotic selection pressure tend to be the type of microbes that we don't really associate with good gut health, but rather often pathobionts, or at least the type of bugs that we might think of as causing opportunistic infections. But through a combination of microbiome and antibiotic resistance profiling and application of machine learning approaches, Molly was able to show that by training random forest models on microbiome samples available before treatment, she could predict the outcome of treatment in terms of microbiome diversity based on only a few variables, in this case, 2 antibiotic resistance genes and 2 bugs. And so what this says is that even though there are these acute damaging features of antibiotics in the microbiome, maybe there's a tantalizing future possibility that we can use microbiome-based diagnostics to understand how the context that is the microbiome might tell us how antibiotics might impact it. So with that acute information in hand, we wanted to ask, what is the potential long-term impact on the microbiome of these kids, especially after they've left the neonatal ICU and they've got other environmental factors come into play. So to answer these persistence questions, again, teaming up with Barb and Phil, but now with work led by Drew Gasparini, a graduate from our lab in the genomics program here at WashU, we had access to a series of outpatient samples from these same kids out to about a couple years of life. And so Drew applied a series of similar microbiome interrogation techniques, now having 3 cohorts to investigate. So what you're seeing here is the number of treatments that these kids get cumulatively. So obviously you've got this heavy antibiotic-perturbed group of preterm infants. You've got the infants who get antibiotics only early in life. And then we added on a 3rd cohort here of term infants born in the same hospital who had no antibiotics at all, sort of our healthy, normal controls. And so through a combination of shotgun sequence to interrogate who's there in the guts of these kids, functional metagenomics, this method allows us to identify both known and unknown antibiotic resistance genes, and some deep culturing and genome sequencing of particular drug-resistant isolates, we were able to begin to answer this question of potential persistent effects. In work that I don't have time to go through, I should also mention that while these human observational studies are very powerful for generating hypotheses, they're unable to actually test hypotheses explicitly. And so one of the things that we're doing now is we're using a series of transgenerational transfer humanized microbiome germ-free animal experiments where we introduce those same fecal samples from this human cohort and then kind of run in replicate the same type of features we see in the human cohort to try to improve our predictions. And that's work that's being led by Drew Schwartz in the lab, follow up on work that Terence Croft and ex-postdoc started. Okay, so what Drew found in this particular study of infants similar to Molly's study, except now with outpatient samples, was some interesting trends in terms of comparing the term kids, who, and this is the resolution of months, the diversity of these kids, as you might imagine, starts very low and then jumps up pretty quickly in the term kids and immediately over the first couple of months sort of achieves the type of diversity you'll see towards the end of the first year of life. Whereas the trajectory for the preterm kids is much more shallow. By 12 months of age, they seem to be at about the same diversity, but it takes them a long period of time. So this sort of developmental delay, if you will, in their microbiome. Of course, the devil is in the details, and I won't belabor each of these stacked bar histogram points, except to say that diversity is not the only measure of importance in terms of microbial composition, such that, we might see, even if the diversity looks like it's similar, you might see features of this type. For instance, here the preterm cohorts, these green bars correspond to Enterococcaceae, and these MDR versions of these particular bugs are completely absent in these term infants, which you might miss if you were only focusing on diversity. And similarly, on the other side, you look at this particular light green set of bars in the term infants, not really present here in the preterm infants, responding to these particular Prevotella. So also showing you that while you might have diversity catch up, you may not have compositional catch up. Now focusing on resistance genes, this other sequelae of antibiotic use, Drew applied this particular technique of functional metagenomics, discovered many thousands of unique resistance genes in the guts of these kids. And interestingly, the point I want to make here is that because he used this method that is initially agnostic to knowledge of resistance genes, it emphasized that even the best resistance databases out there, for instance, this comprehensive antibiotic resistance database, only a third of our genes could really be found by comparison to that database. And whereas all of the genes we found had close homologs, high identity hits in NCBI. So what this tells us is that this method of functional metagenomics is capturing genes that other people have sequenced before, but they haven't identified as resistance genes. So it's telling us that this very clinically important feature is still massively underrepresented in our, at least, database representation of resistance. And perhaps unsurprisingly, when we look at what type of bugs have these particular resistance genes predicted, the top two bugs are uncultured bacteria and bacteria unclassified, obviously not terribly informative there. So emphasizing if you don't know the bugs, you don't know their functions. And in the cases where you do know what the bugs are, they're, again, those type of opportunistic pathogens, not the type of things we want in the guts of these kids. Then we wanted to ask, okay, we know that the resistance is there. How does it seem to segregate across these cohorts of kids who are variably antibiotic perturbed? Interestingly, when we look at unweighted metrics, so that is looking only at the presence or absence of resistance genes and their unique numbers, we find that they're fairly similar across the three cohorts. That is to say, in fact, we might even see a few more unique resistance genes in the guts of these term infants who had no antibiotics. But perhaps now intuitively, you might expect that while you're not changing the sort of playing field of which type of resistance genes you have, what you're really changing is selection pressure for the enrichment. So this is to now say, if you look at the abundance of the resistance genes, they're way higher in the guts of these kids who had lots of antibiotics early in life. So, maybe not a groundbreaking discovery, but something that really affirms the idea that antibiotic use is not inventing new resistance genes. It is enriching from existing reservoirs, and it does so in a manner that is consistent with antibiotic use. Then the other thing that Drew wanted to do in terms of validation was to focus in on these particular bugs that Molly had originally identified as being prevalent and abundant in the guts of these kids. He also found the same thing. This is early in life. And what he wanted to ask was that perhaps early enrichment during antibiotics going to cause these bugs to kind of get stuck in a niche and then still be persistently colonizing them? So what he did was he looked at fecal samples, time matched, or I said age matched across kids, samples taken during hospitalization, and then about 8 to 10 months later, and then trying to culture for each of these type of bugs. And what he found was, in a number of cases, by isolating bugs and then sequencing their genomes, found a number of these bugs that appeared to be identical both at these early and late time points in the antibiotic perturbed kids, but not so much in the healthy infants. So again, now providing evidence that in addition to antibiotic resistance enrichment, you're beginning to see these potential scars, if you will, of early antibiotic use, the persistent colonization enrichment of drug-resistant bugs. Just a quick aside to make sure that we're not over-interpreting this data. This data doesn't say anything about pathogen enrichment. These are not necessarily pathogens. These are just drug-resistant commensals that could potentially cause opportunistic infections given later disturbance in the gut microbiome. Okay, and then finally, to be able to ask whether these features could be detected at the sort of microbiome molecular feature level, what Drew did was he trained a support vector machine, fancy word for a pattern recognition algorithm, on the 16-month samples for the term versus the preterm kids to ask if you were to train a model on these two sets of microbiomes using both taxonomic as well as resistome data, could you distinguish whether those kids were born preterm or with antibiotics? Now, I will somewhat facetiously say that that is a very expensive way to figure out if a kid is born preterm, because you could just ask their mom or dad or a primary caregiver. That's not the deliverable. The deliverable is, if you can do this, it again suggests that there are molecular features, scars, if you will, that distinguish these microbiomes. And sure enough, that's what Drew was able to show. With some feature selection and enrichment, he was able to identify 15 features, roughly half and half resistance genes and particular taxa, through which he could, with 96% accuracy, classify these particular microbiomes. Again, suggesting that there are these features that tell you whether a kid has gone through those early perturbations and perhaps setting you up for interventions in the future. So with that, it's a kind of nice transition towards what can we do if we wanted to perhaps remediate some of these issues. And one such strategy is probiotics. So we've gone in a sense, with what we've shown just before this, and kind of wrecked the microbiome by using the language of warfare rather than the language of microbes in remediating states. So what if you instead use the language of microbes, right? And again, I would argue that perhaps the best examples of this are the very successful use of fecal microbiota transplants for something like Clostridium difficile. But what if we wanted to get a little bit more defined, a little bit more specific with probiotics? So there are a number of recent forays into more defined and engineerable probiotic organisms. And one such organism is a strain of E. coli called E. coli Nissl, originally discovered by a German physician in the First World War as a bug that he found from a soldier that happened to survive a diarrheal attack of the rest of the troops. And so there are a variety of recent academic as well as commercial ventures into taking our sort of favorite model organism E. coli and using all of the genetic tools available to train them on this Nissl strain to have it sort of be a drug delivery vehicle for interesting therapeutics. But there are a couple of challenges along the way in terms of considering how do we treat this bug, which is ultimately a living, breathing, or perhaps breathing, evolvable organism compared to how you might treat a small molecule chemical. So one of the issues to consider is that, again, unlike aspirin or paracetamol, you might end up getting colonization of this particular organism, and you might have it happen variably. So this is data from a clinical trial that shows that after Nissl treatment in humans, up to 24 weeks later, there was still a couple individuals who were colonized with this bug, but it was highly variable across the cohort. And then also, and this is not Nissl work, but in another study similar to the one I showed you earlier, it was shown that a particular probiotic cocktail actually made things worse compared to just autologous FMT in terms of recovery from antibiotic perturbation. So this tells us that, yeah, this would be good to have these defined probiotics, but we need to understand how they work. And specifically, we need to account for their living nature as we think of them as engineerable therapeutics. And so ways in which we can do them, we can specifically control them. We can control things like the expression of the therapeutic payload. And we can also make sure that we can put in kill switches or other types of elements which we can get used to get rid of the bug after it's done what it's done. But in parallel, by using sort of more basic science approaches, I think we also need to understand how these bugs are potentially changing in response to the selection of the mammalian gut through the processes we know all bacteria used to evolve, namely vertical transmission of sort of Darwinian mistakes in evolution that might occur because these bugs are ultimately reproducing and dividing in the guts of these mammals. But also this very, very important complementary method of evolution of horizontal gene transfer, where the bugs, as they're coursing through the gut, may pick up or donate genes with the human microbiome and change along the way. So to do that, we turned back to this method that we've actually used for antibiotic resistance interrogation called functional metagenomics, and instead decided to use it in this case as a proxy for horizontal gene transfer to understand how a model organism like E. coli Nissl probiotic might pick up genes as it courses through the human microbiome to modify its phenotypes. In this case, we made libraries So expression libraries from metagenomic samples, some fecal samples of a group of infants and mothers, created libraries that represent about 10 million unique genes. Those libraries were made by extracting this metagenomic DNA, putting it in an expression construct into E. coli missile, delivering it to mice, and then seeing basically which transformants survived. And then doing this over time and seeing what type of functions we got at the end. And I'm sort of spoiling the thunder here to say that this was a very successful enrichment, because in most of the selections, we went from these 10 million genes to only about 1 or 10 genes surviving in the end, really saying that this selection was powerful. Okay, so this work was all led by Nathan Crook, who was a postdoc in my lab and now is an assistant professor at NC State, and Aura Ferrero, a graduate student in my lab in the biomedical engineering program. So over the next series of slides, what I'm going to tell you is some interesting findings that we got from all of these series of experiments that Aura and Nathan and their team did, looking at a variety of variables. And so because there are quite a few variables, I'll just define them here. We look at the original library, of course, which I've already defined. We vary things like diet. So you'll see that we sometimes use the mouse diet. Other times we use this so-called Western diet, which is honestly just kind of high-fat, high-sugar cookie dough, which I guess is our Western diet. And we do that both on its own as well as with certain prebiotics. And then another major selection variable is what is our library competing against? They can just be competing against each other in terms of monocolonized germ-free animals. They can compete against a defined community of human commensals. They can compete against an antibiotic-perturbed conventional microbiome in animals, or just the conventional group. And so you'll see these icons over the next few slides, and you might also see this, which is the arms of the study, where usually we have the mouse, we're introducing our library, and then we're looking at fecal samples over time and analyzing them. Okay, so one of the first things we observed, especially in our experiment where it was a mouse chow diet, it was monocolonization, so these are germ-free, so just the library, and to see what happened is we saw that our library very, very quickly bottlenecked to select for functions that E. coli missile was using to adapt, that had to do with better utilization of food, right? So essentially a dietary selection. And so what we found is initially through the selection that occurred by sequencing, and so what you're seeing here is effectively particular fragments of DNA that come through and are selected towards the end compared to the million or so that go in. And what we find are these are the contigs that come through from the selection, the bugs that they're likely coming from. And interestingly, in all of these cases, or almost all of these cases, the central gene that is always selected are these, or gene family, are these glycosyl hydrolases, which when we subclone them and then we do a series of in vitro tests, show that they're enabling E. coli to use di and trisaccharides, or even bigger, often would have this glucose or galactose, fructose units, and it's enabling those particular transformants to grow better than the wild type. And I'd also say that in later experiments, when we also did this with fragments that came from our human chow experiments, and when we even just took this so-called Western diet and just made an extract from it, even that enabled those transformants to grow when regular E. coli missile doesn't. So, how do you synthesize all this data? It appears that when the bugs in there, when E. coli missile is in the gut and it has no other bugs to compete with, what it does is it likes picking up elements that allow it to be a better utilizer of the food source that's available. And indeed, when we switch to a different diet, in this case, this human diet with or without prebiotics, the story was pretty similar, right? So yes, there were some other unique fragments that came up, but the fragment that always was selected was these glycosyl hydrolases. Not the same glycosyl hydrolase, but from this family that helps you kind of digest sugars. And perhaps this is not all that surprising when we think of the true ecology of E. coli in the context of other commensals, not in germ-free conditions. We know that even though E. coli has a prolific diet, what it normally does is it relies on the action of anaerobic bacteria that tend to do a lot of the digestion of the more complex polysaccharides that tend to give you the simpler sugars that E. coli likes to munch on. And since those were not around, E. coli was going to adapt now to pick up some of those functions. Okay, Now let's switch gears a little bit and say, okay, that's kind of what we found with our model for horizontal gene transfer. But we also recognize that E. coli can mutate its own genome, and it might do so, I should say, constantly. The genome is mutating through Darwinian processes, and if there are appropriate selections, then bugs might change over time. And I think this is a really important thing to keep in mind, because I would say that, in most experiments, including the ones that our lab does, this is something that people basically ignore, right? You look at most microbe studies and even pathogenesis models, no one is really sequencing those microbes at the end of their experiment. They're just assuming what they put in is what they got out, which I would say, at least based on this, is patently false, right? Microbes adapt, and they adapt very quickly. So we wanted to ask what was going on in this particular situation. And fortunately, we had done all of these controls where we did not have our library. We just had E. coli on its own under all of these conditions. And we could ask over the course of these 5 to 7 weeks, what do these bugs look like? So one of the things that we did was we took about 400 or so of these isolates and sequenced their genomes, and then looked for common mutations that were in mice in different cages and different conditions to identify the type of genes that might be under selection under these particular conditions. I won't go through all of them, but I'll tell you some of the highlights. So one of the things that we find is, again, a parallelism, if you will, in terms of a phenotype that is being selected. And again, that has to do with utilization of food. But now in this case, what we're finding is an example of a gene called NAGC, which is mutated in 10 different ways across almost 20 different isolates and 5 different mice. And what this mutation does is it de-represses the usage of mucin components of this host GLICNAC. And what this really tells you, and again, what you're seeing here are in vitro results that kind of recapitulate this, that apparently what E. coli Nissl is doing is it's mutating aspects of its ability to now utilize a sugar that maybe you don't want it to utilize, right? Which is effectively the inner lining of the gut. Now, again, I'm not claiming like before that this is necessarily turning a bug pathogenic, but it's certainly endowing this bug with the property that we might not think of as useful for a probiotic, or at least maybe useful for the probiotic, but not useful for us. Another mutation, which is in a sense a silly mutation, it's this mutation is a gene called KFIB, which we know is a gene that Nissl has that is involved in its ability to attach to the host gut epithelium. So as it turns out, because Nissl over many decades of its use in the lab had been passaged in test tubes, had never really needed this gene to be around, so it's going to devolve. It had gained mutations in this gene that knocked out its function. And all we found is that it had gained that function back as soon as you put it in the context of the mouse. So again, the reason I said it's a sort of silly mutation is, the only reason we saw it is because it had lost it under these artificial conditions, but it emphasizes how strong the selection pressure is for properties like attachment to the host epithelium. And what you're seeing here again is just in vitro confirmation using cell lines for attachment with and without mucin to show that that is truly what is going on from that gene. And then finally, and this is one that is sort of close to our hearts, in one of our experiments, we introduced E. coli into conventional mice with streptomycin treatment. And this antibiotic knockdown is a very common thing that's done in pathogenesis experiments, but also in experiments that are done with human trials of probiotics, where they initially do this antibiotic washout. Here again, we give streptomycin only at the very, very beginning, and there's no streptomycin afterwards. And yet, in almost 170 different cases, we found mutations to the gene that gives you low-level streptomycin resistance that persisted over the course of our study. So that is to say, even this one single dose of antibiotic in the very beginning gives you selection pressure for your bug to become more drug resistant, which again is something, especially by the FDA and WHO, is a big feature they worry about with probiotic use in terms of picking up resistance. Okay, so now that we've got an understanding of the trajectory of these bugs, I'm going to end on the last couple slides, is what do we do with this information, right? How do we actually make these engineered probiotics do interesting things? And one aspect of that is to be able to precisely control the expression of genes in there from the genome. Now, we know that a lot of great work has been done in getting promoter libraries engineered in vitro, but how do we know what's actually going to happen in vivo? If we do this one mouse at a time, that's a large number of experiments that we have to do with different types of promoters that would sort of run their expression. And so again, now Nathan and Aura, but in a more recent publication, developed a cool new method. And since it's published, I won't go through all of it, but I'll just tell you that it's a neat method to be able to, in high throughput, measure the activity of a promoter library very accurately so that you can then use it as a tool to control gene expression. And effectively, it involves barcoding your reporter gene, in this case, GFP, and then you can mix all of these different variants where the variant is a promoter library in front of your reporter, and then through sequencing of both DNA and cDNA, be able to figure out what's going on. And so, again, I won't go through all of the details of how this is done, except to show that they came up with a pretty nice method that is predictive of how in vitro measured promoter strengths nicely will predict both stomach and large intestine expression, even though it doesn't do such a great job with small intestine prediction of expression. And then finally, they were able to apply this method to actually engineer and deliver a probiotic into the gut. In this case, a probiotic version of Nissl that is able to degrade phenylalanine, which could be an interesting treatment for this condition, phenylketonuria. And they were able to show that both in vitro and in vivo, this was successful at reducing phenylalanine levels in both male and female mice. So I know we're going to try to stick strictly to time. So just in wrapping up, I hope I've shown you that there are these potentials for understanding how bugs and drugs work better if you take context into account. This is how we look at various times pre-pandemic, but this is probably what more look like during the pandemic. I'll end with, again, acknowledging the incredible people in my lab and those who've gone on to bigger and better things, our wonderful collaborators, both within WashU and without, the folks who generously supported our work. And especially in this pandemic, if there's one thing I can leave you with as a PSA, please use antibiotics only for bacteria.
Thank you, Dr. Dantas, for a very interesting talk. Dr. Lampe, would you mind sharing your screen now?
Is that working?
Not yet, I don't think. Well, I think that might have done it. Oh, now I see it. Thank you.
Okay.
Yeah, works. Great. It is my pleasure to introduce the second speaker in our third session of the day, Dr. Joanna Lampe. Dr. Lampe is a professor and associate division director in the Public Health Sciences Division at Fred Hutchinson Cancer Research Center, so our neighbors here in Seattle, and a research professor in the Department of Epidemiology at the University of Washington. She received her PhD in nutritional sciences with a minor in biochemistry from the University of Minnesota, and trained as a postdoctoral fellow in epidemiology, also at the University of Minnesota, before joining the faculty at Fred Hutch. Dr. Lampe's research focuses on the effect of diet constituents on cancer susceptibility in humans, and the effects of human genetic and gut microbial variation on response to diet. Her collaborative studies have involved the application of a variety of omics approaches to characterization of biochemical and physiological response in controlled feeding studies, and to the development of biomarkers of dietary exposure. She has received many awards for her work, including the American Society for Nutrition Mary Schwartz Rose Senior Investigator Award for research on the safety and efficacy of bioactive compounds for human health, and the National Cancer Institute's Division of Cancer Prevention Stars in Nutrition and Cancer Award, which recognizes research contributions in the field of nutrition and cancer. Today, Dr. Lampe will talk about her work involving diet-gut microbiome interactions and influence on controlled feeding studies and metabolic phenotype. So please join me in welcoming Dr. Lampe.
Great, thanks so much, Tomas, and it's certainly a great pleasure to be a part of this today. Many thanks to ISB for organizing this, and to all of the speakers who've come before me with some just amazing studies and results. It's really been exciting. So what I'd like to do today is to take a diet-centric view of the microbiome diet interactions, and to talk about some of the work that we've been doing and some of the approaches that we've been using. in this context. Don't
Dr.
have
Lampe,
to, yeah?
Sorry to interrupt you. Could you change your presenter mode? Right now we're seeing, yeah, I think that might work.
Is that better?
There you go.
Okay,
Yeah.
so I'm gonna have to, I won't be looking at you guys then, so apologies. So from the standpoint of studying diet, one of our big challenges is that the human diet is extremely complex, and there are thousands of compounds that are part of it. And when we think about how we study diet and also how we eat our diets, foods are very complex mixtures. And we often assess them by looking at individual compounds or individual foods and classes, but we really are eating something more along the lines of dietary patterns and the totality of diet. And the whole field of nutrition is actually coming to embrace this more and more, as we recognize that although we've taken a very reductionist approach in the past, following one nutrient, one compound, that really, because of the interactions between all these dietary constituents, we have to think more broadly. In certain populations also, dietary supplements are a huge component of dietary intake, and so we certainly can't ignore those either. So today we've heard a number of different research approaches for studying your gut microbiome, and certainly this comes into play in relation to the diet-gut microbiome interactions too. We've heard a lot in relation to a lot of the in vitro and even the pre-in silico bits before that, as well as the animal models. And the piece that I typically fit in is in the area of small-scale controlled interventions, what is often termed human mechanistic studies these days, to contrast that with clinical trials, which often are related to understanding a disease outcome or a therapeutic outcome. Here we're really just trying to understand the biology. And from the standpoint of the kinds of things that we can ask, certainly mechanistic questions, focusing on particular nutrients, compounds, as well as the microbiome variation. And then on the other end of the spectrum, when we start to get into large population-based cohorts, where we rely more on observational studies, we're really looking more at the broad systemic effects, food and diet, and again, but we can still apply the variation in the microbiome to these studies. So many of the randomized clinical trials would fall into this individual and community-based interventions, which tend to be less invasive on study participants. A lot of what we do in our controlled feeding studies and the human mechanistic studies put a lot of demand on our study participants. So just going back to general gut microbial metabolism, I think it's always good to remind ourselves of where this is happening and what the impact of the anatomy of the human host is on what the microbes see. Thinking about it from the standpoint of how we're designed when we eat food, really human digestion happens before any of the microbial digestion occurs. So a lot of the enzymatic releases within the small intestine are handling a lot of the macronutrients, carbohydrate, protein, and fat. And so it's really the leftovers that become the substrate for the gut microbiome. I mean, certainly down here in the lower part of the small intestine, you do have microbes, but the major action certainly is within the colon. So thinking about it from that standpoint, the microbial substrates are really the components of our diets that we can't digest and absorb and end up and down in the lower gut. And as Thomas Gurry pointed out, you know, that's highly variable. If you're eating a huge load of protein, you may find that there are actually a lot of proteins and peptides that end up in the colon that become substrate for amino acid fermentation. But it really means then that a lot of the reactions that are carried out by the gut microbes relate to reactions that we don't have the enzymes to carry out. So we've talked about fermentation already today, denitrification, sulfate reduction, capacity to break a lot of the ring structures, for example, in relation to a lot of the polyphenol compounds in plant foods, as well as a lot of hydrolysis and deconjugation reactions. So these are sort of the mainstay of a lot of the microbial action. The reason I got interested in this was the National Cancer Institute started an initiative, almost 30 years ago, to understand better the impact of a number of dietary components that aren't considered to be essential nutrients, but may actually affect risk for cancer. And there were a number of phytochemicals that were recognized as their potential role-playing effect. And it became apparent that many of these compounds actually are heavily metabolized by the gut microbiome, such that exposure to them can vary highly across individuals. And it was the lignans that really caught our eye from the standpoint of these being associated with high fiber foods, and we were interested in pursuing this area. At the time, my group was very interested in conducting controlled feeding studies to look at effects of diet on various biomarkers of cancer. But as we recognized, even when we controlled the diet really well, there was a huge amount of variation in response. And we started out by looking at host genotype diet interactions, and then really realized, particularly in relation to the phytochemicals, that it's really the host microbe diet interactions that probably warrant more attention for a lot of these effects. In the context of the plant lignans, there's been a substantial amount of work that has been done to understand their impact in relation to chronic disease. A lot of prospective cohort studies, case control studies of colon and breast cancer, a number of cardiovascular diseases, suggesting a reduced risk for these diseases. Experimental human studies showing changes in things like hormone metabolism, inflammation, and concomitantly with this, experimental animal studies, again, supporting both the effects on these mechanisms, but also the outcomes in relation to tumorigenesis. So lignans can act through a variety of different pathways. This is the case with many phytochemicals when you come to study them, unlike certain drugs that are often targeted for a particular pathway. We find that many phytochemicals, and lignans in particular, can interact through a variety of different receptors. And in some cases, they're acting as agonists, for example, through the NRF2 KEAP1 pathway, affecting a number of the antioxidant enzymes and detoxification systems, biotransformation. Also through estrogen receptor mediation, PPAR gamma, and the RXR receptors. And also they can act as antagonists and compete for interaction with some of the other receptors, and similarly, consequently affect a wide range of processes in the human body. So when we look at where these compounds are found, these are diphenolic compounds. There are a number of different flavors of them and different compounds themselves. Many of them are present as glycosides, and they come from plant foods, predominantly those that are very high in dietary fiber. So a lot of the whole grains, nuts, seeds, fruits, and legumes. And so it's really this component of what we're feeding in the context of a diet, but what actually is going on. And that is related to the gut microbial metabolism of these compounds to two major products, enterodiol and enterolactone. This is starting with secoisolariciresinol diglucoside and metaresinol glucoside, and they undergo a number of enzymatic steps, including hydrolysis of the glucosides, demethylation, dehydroxylation, to produce enterodiol. And enterodiol has been the compound of most interest because of its bioactivity and potential effects on health. But in some of the initial microbiome work that's been done, they've identified some bacteria that can carry out all of these reactions. But for the most part, this requires a consortium of bacteria to allow for the production of enterolactone. And in a study a while ago, but which actually shows really nicely, is just the variation in how individuals, bacteria are producing the end products. You have some, this is a pharmacokinetic study where individuals were given secoisoerythroresinol diglucoside, or SDG as we call it. And you see that this person produces copious amounts of enterolactone with some enterodiol. You've got some people who aren't really making much of either of them, again, all based on the same dosing of substrate. Again, somewhere it's more enterolactone versus enterodiol, and somewhere who aren't making any enterolactone but are producing quite a bit of enterodiol. So again, really dependent on the microbial community as to what the end products are. So we were interested in embarking on a study to look at the potential effects of enterolactone on a number of cell signaling pathways in the colon pertinent to colorectal cancer risk. And started by selecting a plant lignan supplement to allow for conversion to the enterolactones, enterolactone and enterodiol, and then to look at downstream effects in the human host as a result of this intervention. And this study, which was called the Flax Effect Study, was a randomized placebo-controlled crossover intervention in 40-some healthy men and women, relatively young population. Our goal here was really just to understand the basic biology of response in the context of healthy individuals. And we provided a dose of 50 milligrams per day of SDG for 2 months. And the major outcomes were gene expression in colon and rectal biopsies and exfoliated cells. We had participants come in for sigmoidoscopy, and we would take colon and rectal biopsies at the end of each intervention, 2-month intervention, and then stool samples for microbiome interrogation. We also measured 24-hour urines to look at enterolactone excretion. So enterolactone produced in the colon is well absorbed, undergoes conjugation with glucuronide and sulfate in the liver, and is then rapidly excreted in urine. And so looking in these 24-hour urines in the context of the squares, which was the lignan supplement, and the placebo in the triangles, we set this up so we went from lowest excretion to highest. And as you can see, there's a wide range in capacity of individuals to produce enterolactone. Some individuals on the supplement are not producing any enterolactone at all. Others are producing lots of it. And even you see this too in the context of the placebo, where people are exposed to some lignans as part of their usual diet, but even then the amounts are relatively low. So it was based on these differences that we actually split a lot of our analyses between those individuals who fell at the bottom half of ENL excretion and those who were in the top half for many of our comparisons, and I'll share some of those data. So this is looking at gut microbial response to the supplementation. We collected stool samples at the end of each of the 2-month intervention periods, and this was 16S analysis, so pretty much at the general level, looking at the community at the end of the 2 time points. The placebo is the circle and the lignan period is the star. And we didn't hypothesize we would actually see a shift in the gut microbial community as a result of the intervention. This is 50 milligrams of SDG is a relatively small perturbation in the context of habitual diet and substantial fiber intake. So we weren't really expecting much of a shift, but you do see that there are some shifts within an individual when they are on the 2 treatments. And what's more interesting in the context of this is also overlaid with that is their urinary ENL excretion, from dark blue being very low excretors to the yellows being high excretors, and that the overall communities tend to differ between the low and high excretors. So you have a lot more of the yellows and light greens over here and the darker ones over here. But certainly a lot of overlap, which isn't too unusual in human studies. The other thing that we did then was to look at colonic mucosal gene expression using RNA-seq in the biopsies from these individuals. And although we didn't really see a major impact of the intervention itself on changes in gene expression with the SDG supplement, what we did see is that when you look at differences in expression between the low and high ENL excretors, there are a number of things that come up. Many of them relate to inflammation. For example, number of anti-inflammatory upstream regulators, including TGF-beta1 and IL-10, were suppressed in the low ENL excretors. And similarly, using the fecal exfoliome, as we call it, and this is actually epithelial cells sloughed into the stool that you can actually use RNA-seq on a sample here. This is work of Rob Chapkin at Texas A&M, who has established this method, which is really nice from the standpoint of not, in the future, not having to go in and take biopsies, but rather just rely on the stool samples to be able to look at what's going on to the gut epithelium. And in this case, we find, again, comparing the low and high ENL excretors, that we see an upregulation of NF-kappa B and NOS2 among the low excretors, suggesting that there's a differential effect of having the capacity to produce ENL versus not. And it doesn't necessarily mean that it's the ENL that is doing it, but it's really the microbial community associated with that phenotype. So this is work that we're following up on with a goal to try to understand this better. As I indicated, plant lignans are really an integral part of high fiber foods. And so we start to think about the totality of diet and the combination of both the high fiber foods as well as the rest of the diet. We've taken to designing studies that actually allow us to look more at dietary patterns rather than specifically focusing on individual foods. This is a study that was conducted to design two different diet patterns, one that was high glycemic load and one that was low glycemic load. The low glycemic load diet, which the idea is that you're picking foods that result in a low area under the curve for glucose response as a result of feeding. These tend to be foods that are high in whole grains, legumes, fruits, veg, and tend to be low in refined grains, added sugars. And in the literature, we know both from the epidemiologic literature as well as a number of interventions that diets that tend to be higher in these foods, and this is, as a result is why a lot of the public health recommendations focus on encouraging higher intake of these, is that they're associated with the reduced risk of a number of chronic diseases through effects on inflammation, metabolic regulation, et cetera. Whereas a diet that's high in refined grains, added sugars, and more high glycemic load foods are associated with metabolic dysregulation. And a number of studies had suggested that these high glycemic load diet may also be contributing to higher risk of several cancers. So it was this that set us out to do a controlled feeding study to examine this. And we're very fortunate at Fred Hutch that we have a shared resource that provides us with a facility to conduct interventions in humans. We have the Human Nutrition Lab, which is essentially a commercial kitchen and dining area where people can come in and eat on site, and we can provide them with all their food for the span of the study. As you might imagine, during COVID, things are a little different. Nobody's in the dining room, but we are delivering food to participants to be able to allow some of these interventions to continue. Similarly, this facility has a exercise lab, which allows for exercise interventions too. So what these controlled feeding studies allow us to do is to control the diet for the time period that the participants are involved in this. And so for this high and low glycemic load diet, this was the approach we took. The one thing, of course, a study like this has its limitations, and certainly you can't carry on an intervention sufficiently long to allow for disease outcome as an endpoint. And so you're really relying on intermediate biomarkers to monitor the effects of the intervention. What a controlled feeding study allows you to do is to control both what you're intervening with, but also allows you to control the background diet. So often in nutrition, you think, okay, I'm going to give somebody this food, but giving them that food without taking into consideration the context in which they're consuming it, i.e. what other things are they eating, may actually not give you the information that you're trying to obtain from your intervention. So this approach where you can really control diet to the max helps. You can design these diets to test dose response, and then typically the intermediate markers of disease susceptibility are your outcome measures. Also useful from the standpoint of developing markers of dietary exposure, where you can come to understand, okay, you feed somebody a diet high in cruciferous vegetables, i.e. broccoli, cauliflower, cabbage. What does their metabolic profile look like from the standpoint of the kinds of compounds that they're producing, things showing up in their urine, things showing up in their blood. So useful for that too. So in the case of the study that we conducted of low and high glycemic load diet patterns, this again was a randomized crossover design. To the extent that we can, it's often really nice to have individuals serve as their own control compared to giving one group of people one diet and one group the other, particularly in the area of the microbiome where there is so much heterogeneity. Understanding how something works within an individual is really important. So taking this tact has been our usual approach. Again, healthy individuals, 18 to 45. And the diets were designed to provide same number of calories per day for an individual on both the high and low glycemic load diets. Similar macronutrient distribution, that is 50% calories from carb, 15 from protein, remainder from fat. And then as a result, selecting the carbohydrate containing foods, you're really looking at differences in carbohydrate quality, also substantial differences in fiber content as a result of picking foods that are low glycemic load, which tend to be higher in fiber. They also tend to be higher in plant lignans compared to a high GL diet, which low in fiber, but actually not, it's actually substantially higher than the usual fiber intake of the US population. I think our fiber intake is estimated at a mean value of about 15 grams per day. 28 grams per day is actually well within the recommended range, and lower amount of lignans. And then the main study was actually designed to look at effects on metabolic and inflammation biomarkers that had been associated with cancer risk, just to show you that the diet actually did have an effect with regards to glycemic response. The open squares are the low glycemic diet, so i.e. glucose area under the curve is lower with a low GL diet versus the high GL diet. And similarly, insulin response is lower on the low GL diet. So we followed this up with additional study, given that we had samples collected as part of this, to examine the responses to the diet in relation to taking a proteomic, metabolomic, and microbiome approach. And so we had bloods collected beginning and end of each 28-day feeding period. This was a crossover design, so after a 1-month washout in between, the individual received the diet they didn't get in the first part. So again, the contrasts were within an individual. And looking at the proteomic response, we see a number of different proteins differentially expressed as a result of the low GL versus high GL diet. In this case, these are proteins that were expressed. This was using an antibody array approach. So a number of proteins that were lower on the high GL diet here, and then the red is higher on the LGL diet. Number of pathways relevant to DNA repair, replication, post-translational protein modification. Interestingly, we actually recruited the participants in the study on the basis of percent body fat, and there were a number of responses that differed based on whether or not an individual had a high fat mass versus low mass. As you see here across the two columns, in some cases the responses are similar, but for some of these they differ depending on percent adiposity. So in relation to the metabolomic analysis, and I think this is from a microbiome standpoint more interesting even, is that we saw a number of metabolites that differed by diet. This was a targeted metabolomic approach, relatively small number of metabolites that we were monitoring, but we identified several pathways that differed between the two diets, including tryptophan metabolism, branched chain amino acid metabolism, and trimethylamine production, all of which decreased in the context of the low glycemic load diets. And looking at these responses in the context of C-reactive protein, a measure of inflammation, we see too that many of the concomitant changes in both these metabolite pathways as well as CRP were going in a similar direction. Again, not necessarily causal, but providing us a way to move forward on interrogating these further. One of our questions was, given that a number of the metabolites that we were identifying in relation to these outcomes are microbial metabolites, what were the effects of the diets themselves, in the case on secondary bile acid production and the relationship between enterolactone production and the plasma bile acids. As we've heard today a number of times, the primary bile acids produced in the liver, cholic acid and chenodeoxycholic acid, are converted by microbes to deoxycholic acid and lithocholic acid, predominantly in humans. And these compounds have, as well as serving a purpose with regards to lipid absorption in the upper gut, really have become to be recognized as signaling molecules for a number of different conditions. So looking just at plasma bile acids using a targeted platform of 29 bile acids, we see interestingly higher plasma bile acid concentrations in relation to the low glycemic load diet. I would have hypothesized the other way around, but evidently aspects of the diet are contributing to both increases in the primary bile acids, the cholic acids, conjugated forms, as well as lithocholic acid. Now, good to keep in mind that the fat content was, the same on both these diets. So it's not necessarily that you would expect that a higher fat content causing more release and potential conversion to lithocholic acid. But again, a lot of things still to be worked out on that. In this case, in this study, when you look at lignan intake on the two diets across the individuals, you see that there's a huge range in enterolactone excretion, but it doesn't correlate with the lignan intake. So again, here, the microbial conversion is such that there is a huge range in actual production of enterolactone. And looking at enterolactone excretion in relation to those bile acids, we see that an analysis of association analysis, we see that higher enterolactone excretion is also associated with higher bile acid concentrations for a number of these bile acids. Some follow-up work that we've just done recently looking at some of the enzymes involved in enterolactone production by the gut microbes, that many of these are tracking with some of the same enzymes involved in secondary bile acid production too. So some of these overlaps of the capacity to produce both enterolactone as well as some of the secondary bile acids likely come into play here. So just to summarize and to bring us back to where we started in the context of diet and the microbiome, coming at it from a nutritional standpoint and diet-centric view of the universe. And we think about diet and we think about why we eat. A lot of it is for fuel, as well as many other components of diet that are essential for structure and function of the host. In this day and age, certainly a lot of our diet has resulted in energy excesses, contributing to many different chronic diseases, as well as excesses in some of the dietary constituents having this effect. And I think the nice, exciting part about the discussions today is also thinking about the interaction with the gut microbiome. Like if you come at the world from a gut bacterial or gut microbial centric view, you're thinking about what is the impact of the gut bacteria on disease risk and host health. But we all have to remember to keep both of these pieces in mind, both with regards to the effect of diet on the gut bacteria, as well as the gut bacteria effect on the metabolism and exposure of the host to these. So it's an exciting time. Having started at this before any of these molecular tools were available to even interrogate the microbiome, it's really been fun to be a part of this. I'd just like to finish up by acknowledging the support from NCI that we've had over the years and the Fred Hutch, and really to thank Meredith Huller, who's a microbial ecologist who has worked with our group for some 15 years now, helping to spearhead some of the microbial work in this area, as well as Sandy Navarro, who does a lot of our metabolomics analysis in collaboration with Dan Raftery and Ali Shoje at the University of Washington. As I mentioned, Rob Chapkin has developed a lot of the exfoliome RNA-seq analyses that are allowing us to conduct more and more interventions without having to go in and take biopsies, which is simplifying life, as well as other collaborators at the Hutch and around the world. And with that, I'll stop and thank you for your attention. Ready to help answer questions.
Thank you, Dr. Lampe. That was a wonderful talk. We will now have a joint discussion session with both of the speakers. So it's open to questions. I see there's several questions that were asked throughout the presentation. So maybe I'll start going one by one. The first question is to Dr. Dantas, and this is from Priyanka Baloni. In addition to the preterm and term infants, was a corresponding study done on the gut microbiome composition of mothers to say if there's an influence of mother's microbiome on their child?
Yeah, that's an excellent question. In this particular study that we published, we did not look at maternal samples, though we had access to them. We have subsequently sequenced the samples from all of the moms corresponding to these kids, both right at birth as well as a couple times, I don't want to say 6 months and maybe a year later. And we've also sequenced now samples out to 8 years of age from many of these kids. It appears that there are contributors from the maternal microbiome, but the kids' microbiome, and especially resistance genes very early in life, appear to be distinct. So there are some connections to the maternal microbiome, but at least that heavy enrichment we see very, very early in life, for instance, in the preterm cohort, does not appear to be resistance genes from fecal samples in the moms. We really think if we look at the identity of those bugs, most of those bugs in the hospitalized infants are probably bugs that are circulating in the neonatal ICU, the type of things that contaminate surfaces, and you just happen to be able to live also in the gut. So that's the story so far.
Thank you. The next question is for Dr. Lampe from Reba Paul. Dr. Lampe, have you looked at the oral microbiome and its connection with the gut microbiome and its impact on digestion and some of the dietary biomarkers you were looking at?
No, we have not. I think that's a great question and certainly adds another dimension to the story, but haven't delved into that area at all.
Thank you. The next question is from Noah Rappaport, and it's actually closely related to both Noah's and my work on the relationship between the metabolome and the gut microbiome, and it's for Dr. Dantas. Has anyone looked at blood fecal microbial metabolites by measuring coupled metabolomics data in term versus preterm kids to study metabolic capacity loss in more detail?
Yeah, that's an excellent question. I'm not aware directly certainly we haven't done it in our cohort for this particular set of questions. We have started doing a little bit of sort of, I would say very, very rudimentary proteomic and also cytokine profiling of fecal samples to try to understand maybe host-derived aspects that could correlate. I should have mentioned, even though all of the work I showed you was from this antibiotic lens, the preterm infants also have a series of really interesting pathologies, devastating pathologies like necrotizing enterocolitis. So we're also interested in diving into that core to understand whether, much like the antibiotic manifestations, whether there are other changes in the microbiome that will be prognostic of outcome. And that's where I think we're beginning to see some host features. But that's a long way of saying that no, we don't have direct blood or fecal metabolites. I think that would be very cool to be able to look at, but something we haven't.
As somewhat of a follow-up question to that, and you touched upon it briefly, but now that you followed some of these children for a few years, can you pinpoint, or it's a complex study design since preterm birth has its own complications and long-term risk factors, but this repeated antibiotic exposure in the first few days or weeks of life, does it actually correspond to phenotype years later in childhood?
Yeah, that's an excellent question. I'll give you the short answer first. And we just don't have the resolution because of all of the other variables you've mentioned to be able to say, okay, this phenotypic host consequence can be directly linked over to this perturbation early in life. What we are able to do is still ask these questions of, is there still divergence later on the microbiome side? Many years later, now not only related to antibiotics, but we've also analyzed a pretty rich dietary histories of all of these kids. And so we've got, high resolution information about, formula versus maternal breast milk versus donor breast milk. And we are beginning to see very surprising trajectories, not only of the microbes, but we've switched over, and data I didn't show here, but to do genome resolved assembly to get strain level resolution of what's going on in the microbiome as it evolves. And we're beginning to see SNP signatures even many years later that happen to correlate, not just correlate, right? We don't know causation with specific dietary bins of these particular kids very early in life, right? So those are kind of tantalizing for us to understand. Or maybe there was a selection for some sort of, nutrient utilization that got carried over. We'll need to still check that out. But in terms of actual host phenotypes, I think, at best we might get weak correlations, but we'd have to, for me to trust the data, we need to do something mechanistic to follow up on that. And I think our animal models get a little bit close to that. But the difficulty is our animal models, I think, are really good for looking at acute changes. They're not great, obviously, for, there are no animal models that are necessarily going to tell you what happens 8 years later in a human. So we're trying to connect the dots. I think to be able to do the phenotypes, it's going to have to be quite associative initially rather than mechanistic.
Thank you for your answer. The next question is for Dr. Lampe, and it's from Jean-Philippe Gourdin. Dr. Lampe, did you look at the change in the urinary microbiome along with the urinary lignans?
We didn't in this study. We have used, haven't used anything with the urinary microbiome from the standpoint of our interest mostly in what's happening in the gut and the impact on production of particular metabolites. But from the standpoint of being a healthy population and the kind of level of microbes in the urinary tract and the bladder, it seems unlikely to be major contributor to urinary metabolites. When we measure, for example, the lignans in urine, we measure total lignans, so we're measuring both the conjugated forms as well as the aglycone unconjugated forms. So if there's any, for example, hydrolysis of the glucuronides in the urine, then all of those would still be picked up as a response. I don't know if that helps to answer the question.
And I had one follow-up question that came to my mind, and I think it's one that you might be asked a lot. But with a lot of these phytochemical compounds being metabolized by the gut microbiota into potentially more bioactive compounds in some cases, or at least more bioavailable in some cases, is there feasibility in kind of circumventing the gut microbiome and providing the metabolites themselves to individuals as either preventive compounds or the treatment compounds?
Yeah, I mean, that's sort of the first thing that comes to mind, that you think, oh, why bother with all this conversion stuff? Let's just give them the compound. And I think, you know, for some things that may make sense, but I think we need to know more, because a lot of it may be the impact of the interaction of the substrate with the microbiome that is having effects on colon response, either through, you know, receptor-mediated signaling in the colon. Whereas if you were to give those compounds up front, you'd have to make sure that they were enterically coated so they didn't pop out until you got to the colon, because otherwise they'd be absorbed small intestine and really not have any impact lower down. So I think until we understand how are these compounds acting, and, you know, is the systemic effect actually mediated through the compound being in circulation or through the interaction of that compound with the gut epithelium and all the pieces there, then we may be selling ourselves short by trying to just go directly to, you know, a bioactive, so to speak.
Thank you. The next question is from Bilal for Dr. Dantas. Do intestinal microbiota become resistant to dysbiosis induced by repeated antibiotic dosage with passage of time?
Yeah, that's an excellent question. We don't know for sure. We have evidence that, you know, repeated exposure will enrich for antibiotic resistance genes. And it's something we've grappled with quite a bit, at least sort of philosophically, that we tend to paint resistance as always being bad. Right, it compromises antibiotic, usage against pathogens. It could exacerbate horizontal gene transfer issues. But on one hand, at least in the context of a commensal, if you could enrich for antibiotic resistance in a way that doesn't help donated pathogens, that might actually not be a bad thing, right? Because now you're encoding another resilience factor in. We think that there is certainly enrichment for those factors. Whether that makes you more resilient to dysbiosis, I think theoretically it must, right? Over a period of time, you know, to repeated challenges, if you tend to get, you know, microbiomes that don't shift as much. What we don't know is whether there is something inherent about particular people's microbiome states that makes them more malleable or more rigid, right? And there may be so many factors at play: the status of your immune system, how your original microbiome and how the niches got set up. And so we are conducting a series of longitudinal studies, or analyzing them at least, especially when people jump into these pretty dramatic shifts. So, you know, the NICU with antibiotics is one case, but we have a number of studies now getting towards their conclusion of travel, right? So you take people from one country and then you see what happens when they go somewhere else and come back, or, you know, high resolution sampling while they're actually on vacation, and then compare that to the local population. And then you can see whether just observationally particular microbiomes quickly and shift based on exposure to the local environment, or other ones don't shift as much. And then do those correlate with, for instance, in high infectious disease burden settings, of kind of colonization with the local bugs, right? And I think that helps somewhat answer this question of, through repeated insults, whether it's antibiotics or other bugs, will they change? Almost certainly they will. The question is whether that's a good or a bad thing, And could you, most importantly, predict that a priori, right? And I think we're getting better at doing that, being able to say, okay, you give me your starting microbiome state, maybe actually even give me, now this is harder, but give me 3 or more status updates of your microbiome before you travel. So I can get kind of a trajectory of what your actual variation is, and from that, give you a kind of resilience index. And we're far away from doing that very well, but we're beginning to get there.
Thank you. The next question is from Sean Gibbons to Dr. Lampe. What are your thoughts on whether or not the human gut microbiota modulates energy harvest from diet, and how this may interact with obesity?
Yeah, that's a great question. And there've been a lot of studies done looking at fermentation capacity and therefore production of short-chain fatty acids. And, even just thinking particularly in the context of very high fiber diets, that some of the very old nutrition studies came up with estimates of how much of the diet was actually attributed, or energy was attributed to the activity of the gut microbes, and it could be anywhere from 10 to 15% of calories were coming from contribution by microbes. But I think, a lot of this also has to do with the signaling aspect of this. And although, yes, you can provide more energy, the effects of the short-chain fatty acids on a lot of the metabolic signaling pathways is probably more important in the context of how the microbiome is contributing to energy harvest in relation to across individuals than particularly the specific fiber microbe interaction per se. I mean, it really relates more to how this is impacting the host response to the various compounds. And often it seems like you hypothesize that it should work out this way. And often what we're seeing in the context of some of these studies, at least, to the extent that you can conduct them in humans, that it's showing the opposite. So I think, going back and forth between the mice and the humans to try to work this out and to try to work it out in the context of very controlled studies in humans is a direction that we need to go.
Thank you. The next question is from Lauren Anderson for Dr. Dantas. To what extent is the microbiome diversity differences between preterm and term infants due to C-section?
Yeah, excellent question, because there has been a lot of work on this. So I would say in our preterm cohort, at least when we looked at those acute differences, we did not observe any differences in the composition of the microbiome based on mode of delivery. Now, I think there are a couple reasons to expect that. I mean, again, hindsight, maybe explain that, is, again, just that the low diversity enrichment of those particular types of bugs, right? So I think when people have shown differences in mode of delivery and the microbiome, you're usually looking at kids who are very quickly then exposed to, other features. They have sort of more normal trajectories. And so there it makes more sense that you might have vaginal microbes versus skin microbes that dominate. None of those things I should say, any of those impacts are just wiped out by the fact that you've got this massive antibiotic selection pressure and a much more constrained environment. So that's how at least we explain it. We see no differences there in the preterm cohort. In our term cohort, we do see some influence. So this is, again, now no longer comparing term to preterm, just looking within the term cohort. And that is an interesting cohort, because I didn't mention it for the purposes of this, but that is a twin cohort as well. So all of those kids, there's a co-twin. And so we have an environmental and a sort of genetic control there. And we're taking that into account as we look at, especially, right, one twin got a different diet, for instance, because one was breastfed for a period of time, or got an antibiotic. And how does that change the trajectory that was maybe imprinted from mode of delivery? So the short answer is no impact in our preterm cohort, and sort of a minimal, statistically significant but low magnitude signature just early in life in our term cohort.
Thank you. Dr. Lamb, the next question's from me. I was hoping I can sneak it in, but one of your results showed variable changes in blood proteomics in response to a low glycemic diet based on adiposity. And I was wondering if you have any thoughts what might be driving this interaction effect.
Yeah, you know, we see that quite a bit, that individuals with a normal percent body fat are often responding very differently than those with a higher adiposity. I think part of it is where you're starting in relation to many of your metabolic processes, where you are in relation to kind of low-grade systemic inflammation. So how things shift can be quite different. And so it seemed like for some of the inflammation stories, for example, that the responses that we would see with the effective diet, we would see, for example, in the context of those individuals with higher adiposity, where we wouldn't see an effect in the low adiposity individuals. But things like some of the signaling in relation to metabolism, the normal percent body fat individuals tended to be more responsive to the dietary effects, and you know, we're able to shift and move. And we see that too also with some of the bioactives, some of the phytochemical bioactives, that we often see effects of these in individuals of normal percent body fat, that in the context of obesity and overweight, that often the response is blunted. So many of these, I think, sort of relate to, you know, what is your basal physiology and milieu on which these compounds are acting.
Very interesting, thank you. And we're close to approaching the end of our discussion. Dantas, I had one more question for you, and it involves your work that you present at the very end of your presentation that you briefly touched upon, is the engineering the probiotic to be actually able to degrade phenylalanine, and as a potential treatment for phenylketonuria, for example. My question, I was curious, how stable with this approach is the functionality of that probiotic long term?
Yeah, excellent question. I think I had like one little quick blurb on it that I didn't get to. So we were able to, because we were, you know, a large part of that study was looking at colonization dynamics and stability of genotypes. We did sequence those particular engineered probiotics too. In that case, out to a couple weeks, and we did not observe any mutations in the actual engineered components. While we did observe random mutations come up to the genome at approximately the estimated frequency and the measured frequency of, you know, just at the regular mutation rate. So just based on that, it appears that at least in that animal model, they appear to be stable enough. Now, you know, that's only one shot on goal, if you will, right? There are lots of other features to consider. And also, I think that the question will really become, how do you balance between wanting to have a longer lived probiotic, so you don't have to force people to take this drug all the time, versus then the longer it stays around before you give another dose, the greater the chance that things could devolve. Mm-hmm. So we did not observe any changes. We saw no promoter changes, no changes in the gene itself, even though there's no other selection for it over the course of those experiments in those mice. The only thing I'll say is in a lot of our experiments, we did observe that Missile has the capacity, much like has been shown by others in human trials, to transiently and somehow variably colonize the gut, sometimes even in bizarre places. Like, you know, we were able to harvest Missile out of certain parts of the small intestine, which you really shouldn't be seeing E. coli sitting around there, right? We don't know how much of that is the idiosyncrasies of these mouse experiments, but it just tells us that even if we don't necessarily see the you know, massive genetic changes that impact the actual gene that we're putting in, other changes in the genome might still cause this bug to behave differently than what we wanted to do. And so that's one of the big reasons why we're designing kill switches, so that no matter what it does, we still have control over its sort of on-off nature.
Very interesting. Well, I think that's the end of our discussion. Thank you both for really, really fascinating talks. We'll reconvene in 3 minutes for Nathan Price and his closing remarks. Thank you again.
Thanks.
Thank you.
Let's see. Am I on? All right. Great. Well, hello. I'm Nathan Price, Associate Director and a professor at ISB. I just wanted to say what a great day of talks. Give a big thanks to everyone that was involved. I want to thank again our sponsors, Metabolon and the Washington Research Foundation. Without you, it would be very hard for us to put this on. And we really appreciate that support that makes this all possible. I did want to thank again the outstanding set of speakers. I thought all 6 of the presentations were just fantastic today. I learned a ton. I think everyone online did. And I also wanted to thank the audience for really great questions and active engagement throughout the whole day. So thank you to our outstanding speakers, our session chairs, the attendees, and the support staff that made this event happen. Overall, I think it's clear that the microbiome as a field has been maturing, that it is gotten to a state, as we heard today, that is beginning to have real predictive power in many arenas. And we thank those that gave us the great talks on computational methods, on in vitro systems, and on in vivo systems as well. And the fascinating health implications that we find, and the ability now to think about how the microbiome can have, the understanding of the microbiome can have real impact on our understanding of health. Also, just in closing, just want to say that we do put these on for free. And if you're in a position to do so, as a nonprofit research organization, we do rely on the generous support of our donors. ISB has pivoted quickly in the wake of the pandemic, as so many of us have, to put on a number of virtual events such as this one at no cost to registrants. And so we would greatly appreciate your support, which is needed now more than ever. So if you're in a position to help with this, please consider making an online donation at isbscience.org/donate. And you can learn more about ISB and the science that we do at isbscience.org. Thank you again so much to everyone for participating with us, for the audience, the questioners, and the fantastic speakers giving us a view of what's happening across the world in microbiome. Thank you.